We will soon begin Bio Korea INVEST 2021 from 10 a.m.
May I have your attention, please? In a moment, from 10 a.m., BioKorea Invest Fair 2021 will begin. This event will be held in compliance with the government's guidelines for preventing infectious diseases in response to COVID-19. Guests are requested to observe social distancing and make sure to wear a mask. And this event will be broadcast live not only on site, but also online. You can also download the company briefing presentation material files and refer to event information through the official website as well. And you can watch both Korean and English page as you want through the official website. And simultaneous English and Korean interpretation will be provided for today's event. Please note that Channel 1 is for Korean and Channel 2 is for English. And also, please leave the interpretation receiver at your seat when you leave. And in a moment, the second day of BioKorea Invest Fair 2021 will begin. So please wait a moment. 네, 장내에 계신 여러분들께 잠시 안내 말씀 드리겠습니다. 지금 테이블에 통역 수신기가 비치가 되어 있습니다. 혹시 통역 서비스가 필요하신 분들께서는 테이블에 비치되어 있는 통역 수신기를 이용해 주시기를 바랍니다. 아울러 사용하신 후에 퇴장하실
And
때
also, when you leave the venue, please place your receiver back on the table. Thank you.
Welcome to Bio Korea INVEST Fair 2021, and thank you for attending this event. I am Son Young Park, and I'll be the MC for today's event. From now, we will begin the second day of Bio Korea INVEST Fair 2021. You can watch both the Korean page and the English page as you run through the official website, and also simultaneous English and Korean interpretation will be provided during sessions. So please note that channel 1 is for Korean and channel 2 is for English. Today, also at the Bio Korea Invest Fair 2021, the pharmaceutical, bio, medical device, and healthcare companies, which are seeking to advance into the global biohealth market, gather together to talk about their own superior technology and visions. So we hope that this event could be the time that everyone participating in this event get fruitful outcomes. Unfortunately, this forum is being broadcast live through the official website as we are unable to invite many participants in order to prevent the infectious diseases of COVID-19. And at the site, domestic speakers, hosts, and managing institutes and partner organizations are participating while observing the social distancing and mask wearing according to the guidelines for preventing infectious diseases. So then we will start the third session from now on. In session 3, there will be a presentation by Youngjun Ko, Head of Discovery Division of GI Innovation, with the topic of new therapeutic strategies for NK cell-targeted immuno-oncology. So I'd like to invite him onto the stage. Please welcome him with a big hand.
Good to see you all. I'm Ko Young-jun from GI Innovation. So I'll begin my presentation from now on. And before I begin, I'll mention that Chang Myung-ho, CEO of GI Innovation, and Lee Jun-heng, Bixabio CEO, will cover the discussion center. So today I'll cover the NK cell targeted immuno-oncology, and this slide is the disclaimer. And first of all, in the global market, we're currently working on NK cell targeted IO therapies and cell therapies and immunotherapy, among others. And I'll briefly cover what status we're currently at and then move on to further details. And as you all may know, in the immuno-oncology market, there are first generation chemotherapy, second and third immunotherapy, and nowadays the fourth generation targeted metabolic therapy is a thing, and immuno-oncology therapy is a hype, and everyone is paying attention. And in terms of it, and there are metabolic therapy, which is gaining a lot of attention. However, so far, immuno-oncology therapy treatment is still gaining a lot of attention. And today I'll cover cell therapy and IO target therapies, and they're specifically they belong to the third generation immunotherapy. And they among other various treatments that target this NK cell, and there are various modulators, including NK cell, and there are cytokine activators, among others. And there are interleukin or other agents are mainly used, and there are T cell, cell, and there's T cell engager, and thanks to the boosting therapy of that, we can use that NK engager among other efforts. And in an hour, the 10 panel discussion will be covered, and also the pain discussion therapy is also a very important key. And I am keep telling about this issue, but CAT-T treatment is also gaining a lot of attention, and it combines with gene and high NK form and other therapies. What if we come up with treatment of such? So this idea, not only in Korea but overseas, are agreeing with this idea. So we're in partnership with other entities. Well then, why is NK cell important, and why is that idea gaining a lot of attention? So in fact, something that can kill the cancer cell really well is the characteristics of this. So for T cell, there is a specific characteristics that this antigen can be recognized this, and it can kill this. And without having to go through that process, there are stress antigens that can be directly targeted and killed by this. And actually, those NK capacity, if you take a look at it, you can take a look at cytotoxic molecules, and they harbor that. And when they meet cancer cells, they can kill cancer really well. So there are various combination with antigen, antibody, and ADC, among other therapies. And in terms of NK cells, one strength is that you can do tailored therapy, and you can expand its use. And for NK cell and T cell treatment, the biggest difference between the two is that low risk. You can work on multi-dose until the cancer cells die. So unlimitedly, you can keep administering this for a treatment purpose. So that is the key strength. And one more thing that differentiates is allergenic treatment. So T cell treatment so far recalls a self T cell, and it has to work autonomously. But for NK treatment, there are advantages for allergenic characteristics, so it can massively increase its dose. So we can prepare them, and we can inject it to patients. So in terms of productivity for a company, there are a lot of benefits. So that is what the key idea. And T cell is in fact related to immuno-oncology and DBTM. But if it kills, it doesn't stick, but it can relate to adoptive image, and it can come up with additional treatment. It can come up with additional impacts. So there are various NK therapy and the optimized immunotherapy can be worked. And what kind of directions do global companies have, and how they develop immunotherapy is shown on this slide. And typically, if I would begin with NK cell, then you can work on health cancer patients or healthy donors. And for cord, you get this umbilical cord blood from a cord baby, infants. So if you categorize this, and if you work on a specific way to proliferate this and use it as a treatment, and in fact, in Korea, there are many frontline NK treatments under development. Get shared of this information and proliferate NK cells and administer that into patient's body. And nowadays there are some news for companies make iPSC, and again they detail in NK cells, and they can manipulate genes and use it as treatment. So that is another option. And NK cell expanding, if you can proliferate this treatment as you want, and there are two main ideas, and whether you use pivot cell or not. And as I said, they're NK, and they're through using genetic engineering, and you have to come. And this is known to have less side effects, but if there's tumor-associated antigen, if you can target this, then you can go conduct engineering and inject this. Or I'll move on to this very soon, but there's the cast. This company uses cancer cells, and NK. You can use NK cell to use it as a treatment. And in terms of IO, there are cytokines that you can use, and of course, it is risky to use cytokine. However, nowadays cytokine is gaining a lot of attention, and a lot of global pharmaceutical companies are paying attention into this, and many global developments are currently ongoing. And in terms of immuno-oncology, there are various IOs are currently used, and there are co-inhibitors or NKG2 1 and 2 are used as targets, so they're specifically used. And in terms of T cell engager, you can use bio-twice specific to use it as a NK engager to highly effectively kill tumor cells. And there are various companies, as you can see, and how and from where, which source you get NK cells is a thing, and they drive this and adopt them. And if you compare the advantages and disadvantages, then the key idea is that it can used off the shelf. So whenever a patient comes, you can just sell it and deliver it. So that is the biggest strength. And no matter what kind of cell source or NK source, you can use it as very usefully. And for peripheral globe or cord blood, you can receive those NK cells from healthy donors. And if you administer that into patient's body, what it helps is that those mature and ready cells can kill those tumor cells. And in very relatively short time, for example, 2 to 3 weeks, and you can proliferate that and administer in patients. However, the problem is whenever the donor changes, the NK's key gene changes, and that is the risk. So how we're going to work on that is the key challenge during the GSP process, and that is needed. And I briefly mentioned this, and there you would use iPSC or natural question MP courts. In terms of iPSC, of course, gene modification is done, and then there will be gene huddles, and there are gene characteristics. And the iPSC's fundamental problem is the teratoma problem. How we're going to deal with that issue is something that is clearly a challenge. And for here, cell court, there are homogeneous characteristics. However, the ABCC efficacy is relatively low, so we need additional gene modification. And in terms, the cell treatments has a certain level of hurdle. Next, there are various developers here, and we conducted some research on them. And as I said, in Korea, there Boxel Bio or GI Innovation or other companies that work on NK cells. They're engineering. They get blood from healthy donors, and you proliferate NK cells well and inject that into patients. Is widely adopted. And other overseas companies use cell treatments that are not engineered, but nowadays Chi and K or other kinds of engineering are done to increase efficacy is currently being worked on. So in summary of this part, the NK treatment itself is a pure NK, not engineered therapy, or a slight a moderate level of moderation is done, and administer that to patients can be done, or immunotherapy is widely developed, and NAT Pharma is developing this frontline, spearheading this industry. And cytokine based is NIF59 was shortly mentioned. In fact, a cytokine based therapy, since nowadays IO industry is gaining a lot of attention, so cytokine therapy is also gaining a lot of attention. And recently, Metatherapeutics NKIT cells recently got 4 trillion Korean won on investment, and nowadays we're investing in GI 101, and this also is cytokine based treatment, and we recently made a global deal. And there are various kinds of NK cell engagers, bispecific or tri-specific characteristics. Of course, we're taking baby steps, but we are taking various efforts, and many venture companies are giving a shot into this sector. And next, I'll briefly cover clinical phases and NKG2D or co-inhibitor. Among other stimulations are being done here, or one familiar idea is the co-inhibitor signal that you may know, simazine, XL, or TIGIT, or other inhibitors are currently in progress. And likewise, there are various other kinds of phases, clinical phases. Another CAR-T therapy is gaining a lot of attention and under development. And NK treatment is nowadays gaining a lot of attention, so there are a lot of clinical trials being done, and the frequency of clinical trials are frequently growing. And in fact, one of the key limitations of the cell treatment is blood cancer. It works really good for blood cancer, but it doesn't work with solid cancer. There were many voices. However, nowadays a lot of clinical data. So if you take a look at it, of course, it works really good for blood cancer, but the NK cell may have efficacy on solid cancer too. So there is research. So we will keep contributing and putting efforts into this IO treatment and NK cell therapy industry. And next slide. This shows visually the entire market, and there was a boom skyrocketing. It shows 129% of growth rate, and if you take top five companies, it shows 129% of CAGR growth rate in total. A lot of global pharma companies are paying close attention, I suppose, because of this growth. Well then, why do global pharmaceutical companies are paying a lot of attention in this industry? Is that why are they paying attention into IO treatment? So in order to answer that, I'll briefly cover this part again. So NK cell itself can have a lot of high IO immunotherapy efficacy. So NK cells can be well recognized and create this, and their innate immunity. So adaptive immunity can be induced as well on top of it. And there are many clinical trials are being done, and they are showing bright potentials and profiles. And unlike other T cells, it can work on targeted treatment, and it is very safe. So that is why the global market is paying a lot of attention into this. So they are paying attention to IO. And NK targeting IO treatment, and there are various goals, and there are huddles, and there are approaches, and of course, making a goal is how well we are going to kill tumor cells and how safe we're going to make that. And if you think of NK cell, then the cost can be really high. So how are you going to decrease the cost? And since we're a pharmaceutical company that develops drugs, we have to decrease costs and how well effectively we can treat our patients is the thing. So we have to take that all of those into consideration. So from cash and potentials. So these approaches. So how are we going to expand this, and how we're going to develop this cytokine-based platform, and how are we going to develop this platform for other Keytruda developed by Samsung or other successive treatments if we take compound administration, among other strategies can be used, and Parexel Bio or among other companies are also taking this approach, an automation system and cash value system among other autonomous system can be used for this. So here there are some representative data, and this GI 101 is the key IO treatment of our company, and currently US MST mark and key licensing is ongoing, and we got approval. So I'll show you this brief results, and we conducted efficacy experiment on monkeys, and this is IL-2 based, and T cells are used to increase immunotherapy efficacy, and also we can proliferate NK cells. In that sense, we can optimize this NK cells, which is agnostic, and we can activate these cells. So that is the basic characteristics. And here I see some glitches here, but those pipelines. Instead of T cells, we can proliferate NK cells more. So that we are currently working on this pipeline as well. And for GINON, compared to other activators from other companies, based on our public information here, our pipeline can significantly increase our efficacy like this. And I also mentioned monalizumab, and it uses NK for anti-cancer, so it can work well in the human body, and it also uses engager. And J-Cell, which is a partnering company of ours, is also working on this NK cell, and we have our own platform to proliferate this and see if we can actively kill cancer cells. And we used this one on the TOFP NK, and it created a pore to kill tumor cells, so we do have that data. And on top of that, in order to proliferate that, instead of Chi-NK, we used chemotherapy to nanotherapy to come up with this platform, so we could detect this research results. So if we combine all the data here, then can we get rid of all the challenges? Then we need to pay some more close attention. And as you may know, cancer does not occur based on one reason, and you cannot use one treatment to treat it. So this recent data was in 2018 from Switzerland, and a pediatrician doctor announced this. There are various treatments, and if you combine various treatments, then you can come up with effective therapy measures. And there are anti-cancer vaccines, cytokine, and NK treatment or immunotherapy. If you combine all of them, then it is very sophisticated, and if you administer this, then it can be worked, and they are effective. And NK treatment or NK targeted IO therapy, we are paying a lot of attention, but I think it needs that we should combine various technologies or treatment like this. So that is it for my presentation, and thank you very much.
Thank you very much. We would like to thank Youngjun Ko, Head of Discovery Division from GI Innovation, for his informative presentation. And he has talked about the theme of the third session, which is new therapeutic strategies for NK cell-targeted immune oncology. So you can also download company briefing material files through official website. So now we'd like to move on to the next turn, which is a discussion session. So I'd like to invite Young-Ho Jang, PhD, founder, CEO of GI Innovation, and Junheng Lee, CEO of Vaxel Bio. Please welcome them with a round of applause. So then, please proceed with the discussion.
Thank you very much for inviting me to this excellent occasion. It's such an honor for my company. First and foremost, it was really interesting to see the data that was presented by GI Innovation, and I think it's better for us to carry out our discussion based on the presentation materials. I think all of you who are here today would be interested in NK cell therapy, and I'm sure you're here and having your interest in NK cell therapy. And I want to ask a few questions for CEO Chang, and then as he answers my questions, we will be able to broaden the horizon of our thoughts. So first and foremost, I want to ask one question. You showed us many different pipelines. There's autologs, and then we what we do is we extract glucophilus from the cancer cells, and then develop that into a therapy. And one of the challenging questions that we get from IR sessions is that if you think about Fate or many different companies in the U.S. that are focusing on immunotherapy, IPS or allergenic or cord blood origin NK, they utilize these AKs and then try to pioneer and have market positioning. But then they ask, why do you want to focus on autologous? What's the reason why you would like to, stick to this plan? And I think CEO Kim, no, I'm sorry, the Discovery part head Kim showed us a great path about why that they are focusing on autologous. And I think it's time for us to think about whether we should just stick to autologous or switch back to allergen. What do you think is a better approach? What is wiser?
Thank you, Dr. Lee and Professor. It is a great honor to be here with you. So, this key idea, especially for IO, many therapeutic and pharmaceutical companies are paying a lot of attention and they're agonizing over this issue. And there are some other companies, they're thinking of how to develop healthy allergenic and NK cell and how they can freeze that and administer that to patients. And we were between autogenic and allergenic therapy. And for this directly extracting that from people, and it can decrease side effects, and we can use that directly. And there are rare diseases and highly difficult to cure diseases, and we have to secure cells and proliferate that and use it as treatment. But there are cases when patients have to go through radioactive therapy or chemotherapy. So there are patients that have so little NK cells. So we have to get healthy donors NK cells and proliferate them and inject that. So in the future, NK cells from healthy donors, how well they will match with patient cells is a thing, and markers among various markers, allergenic cells, and freeze them, and through clinical studies, we figure out the best suitable allergenic characteristics to increase clinical efficacy, and we have to keep working on that research, and also Kaikane, among other ideas, will gain attention, but the problem is the cost. Those disadvantaged people, if they do not get NK cells, and if wealthy people get treatment, then that is also another disaster. And specifically, one another challenge is that there was a paper from Nature. There are transferable cancer cells, and it is the NK cells that can mitigate that. And steady cells, if they get unfrozen, then it can increase cancer cells. So that also gained a lot of attention. And maybe because of time limits, I may not cover that. But nowadays, cancer is not. We are not living in an era of treating it. We are living in an era to prevent cancer. So before you get cancer, you have to prevent it. And if there are dormant cancer cells that can be transferred, then we have to keep mitigating and suppressing it. So you have to bank your healthy cells, and later on, maybe you can inject that to your own body. And if you are older, then maybe you may get healthy and young donors cells. Because if you get older, the higher the risk that you will get cancer. And for that, probably a genetically engineered NK cell, there are a lot of hurdles. Those non-patients or those people who got cancer, I'm not sure if there will be some donors. So let's say, not sure, from 10 million Korean won to 50 million Korean won if we could offer this healthy NK cell. And there will be a market, there will be demand. So for those people who have potential of getting cancer, then they may be customers. So we are also thinking of that, and also allergenic. And nowadays, we recently succeeded in 50 embryonic, and recently we're working on 200 dose as well. So that is my answer. Thank you.
Yes, to be honest, this question is very similar to do you prefer your mom or do you prefer your dad? Because there are pros and cons depending on which option you choose. And there are people who want to make therapeutics utilizing NK cells, and this is basically our homework for everyone. And as scientific technologies are being developed over time, maybe there will be a joint solution for people who are facing individual challenges. And this scientific development will be fed to our dilemma. So, if you are interested in investing in this area, you need to do a deep dive research and study for you to understand. And you might think that there's a challenge, and you might think that all you need to do is to resolve this issue, but that is not the case. We will be facing another round of challenges even after we resolve this issue. And what we do is we screen all the papers and scientific research all around the world, and so far, NK was not considered very promising compared to CAR-T or other technological options. But recently, the reason why it is getting attention back from the industry or people is because a check parameter was included in cancer therapy, and the margin for NK cell has been growing because of that very factor. And instead of just using NK cell, you're going to treat cancer. This idea itself could be too ambitious. But because we have more options and because we know better about cancers, there are, it seems like hurdles are being cleared away from our site, and I think a great result will be driven later. I'll also mention this, and I present a material for my company, but when we develop NK cell based therapy, it's not about we're going to kill the cancers just using NK cells. we're going to use diverse options, and then NK cell therapy could be a very efficient way of treating the cell cancer cells as well. This is our joint homework, and I think what's important from CEO Chang's point is that we need to consider costs. We have to understand how we're going to set the price range for this therapy so that more patients can benefit from the new developed therapeutic approach. In that sense, whether autolog is better or allergens better, we need to think about that from the cost perspective. And because we have many different developing technologies that can increase the efficacy, and especially the ones who are focusing on stem cells therapy, what you do is you extract healthy stem cells when you're young, and then as you develop cancer, when you're older, you can develop therapeutics from your young self stem cells, and that could be an idea too. In that, at that point, whether iPSC, NK cell autologs or allergen, we don't even have to choose these options. Maybe you can extract a little bit of cell from autologs, and then we will be able to be equipped with a technology to use that little amount of cells. And I think more options will come in the future for everyone. And let me ask then another question for you. GI innovation presentation covered that some of the practical challenges when it comes to production and manufacture. It's really hard to compare individual manufacturers' capacities or production methods. But GI is feeder cells to expand NK cells. But what happens in reality is that there are variables when it comes to using feeder cells, and so many complicated scenarios happen, and autoimmune system could meet some challenges and hurdles as well, and that is why so many people are using feeder cell free culture system. They're focusing on R&D projects in this regard. But then what happens is that whenever there's cancer or viral infection, NK cell recognizes those and then it gets activated. And that is why we're using feeder cells, because feeder cells are basically cancer cells. And in order to avoid this step or process, many different technologies are being born, but it seems that you are also trying to avoid feeder cells, the use of feeder cells. And as scientists ourselves, if you think about physiology, NK cells have to recognize some cells, for example, cancer cells, so that it can recognize and try to attack those cells. And that mechanism has to be activated. And when you are trying to eliminate feeder cells, do you have any theoretical background or any way to have a breakthrough? Can you introduce some of the mechanism, please?
Thank you. Probably that question will be the most significant question for today. And in terms of feeder cell, we have thought about how we're going to use it. And this is something that I covered at the last part of my first question. And in the future, there will be a time era where healthy people get injected of healthy NK cells. So we have to decrease the manufacturing processes, and there are exosomes and other genes or other content should be included in the cell. And of course, that technology is very good in terms of conventional technology. And if we thought about effective proliferation ways, and we IPS, and we use peripheral blood, and we tried proliferating it, and it stopped at 100-fold or 160-fold. And that level doesn't meet the commercialization level. So that is why we used IPCC, even though we know that it is risky, and we came up with that. And we proliferate that without patience. And we developed the platform and tried various NC proteins and bi antibody and cytokines, among other combinations, and came up with the most effective way to proliferate it, both in vitro and in vitro, and to find out the best combination. And dual body and cytokine platform. And if we make that, we conducted 50 times and 100 times of experiments, and the NK cell derived from healthy people, and it proliferated to 12,000 or 13,000 folds. And we kept continuing a 50-meter proliferation, and 130 buyer was produced. And even if we continued freezing it and melting it for 30 times, then it was kept its healthy status. So in preparation for the future, if there is no feeder, then that will have more advantage. But for cancer patients, the pivotal delivering NK cells, if that is used, that is rather even more effective, and it can kill cancer cells. So our company's way is to better prepare for the future. So not only in Korea, but including overseas cases, if we increase that by 13,000 fold, then it is very successful. So whenever you visit our factory, then I'll be happy to show you.
Yeah, I think time is almost up. There are issues that are discussed in the industry, and they are all linked to approval and also costs. So I have to make sure that our therapy is cost effective and also safe. No matter how many times we say it is safe, if we fail to persuade the Korean FDA, then it's all meaningless endeavor. As a result, we need to be able to have some evidence to persuade and convince KFDA. And now I think I can feel we need to have this pioneering spirits to challenge ourselves to overcome the hurdles that are present in front of us. And for other companies who are pursuing NK cell therapy, and you're definitely a leader in the market. Maybe I can go to the final question of today's session. A recent article in Nature said NK cells are actually putting tumor cells in sleep. They put tumor cells to sleep, which can invoke future challenges or risks in the future. But that's more of a physiological status. When it comes to therapeutics, it is a different story. GINO1, this material is going to be combined in order to target the problem. But then the problem is NK cells' strength, the virtue of NK cell is safety. Compared to any other cell therapeutics, NK cell is the most safest form. But if you increase this, if you combine these two, then there is a side effect that might arise in the future. So can you say a little bit about this?
So we used SafeIL2, which is safely approved, and it decreased the weight or when we conducted mice or monkey experiments. And as I mentioned, GI101 it increases TI cells, and other in terms of that, NK cell significantly increased compared to the control group. And so we currently developed it and under development. And the key is that, as you said, T cell has to moderately increase an NK cell. And why do we need a CD cell? Is that it has a memory characteristics. For example, if we use compound treatment, then it is very effective, and it creates a lot of antibody. But for CDC cell, it creates moderate level. So we target those treatments, and thanks. The key is the memory function. Thank you. If we keep talking about this, probably we can talk all night. But these ideas are key issues that we are keep thinking of, and so I gave you this question just to bring this up. So please pay your continuous attention. And if there is anyone engaged in the media outlet, if there is someone who would like to know about us, my company or GI Innovation, if you contact our companies, then if you provide us an opportunity to further elaborate. But of course, anyone, any investor here are welcome as well. Thank you for listening. Thank you.
Thank you to our two panelists for their insightful discussion. And they have shared the ideas to develop new therapeutic strategies for NK cell targeted immune oncology. So next, I'd like to invite Young-Jun Ko, Head of Discovery Division of GI Innovation, again onto the stage for his presentation on his company. Please welcome him with a big hand.
Thank you very much. I think I introduced some of the corporate messagings and vision in the first part of my presentation, which was a thematic presentation. So I'm going to be brief on that part. As you can see, our company was established in 2017, and we have about 69 employees working at our company. As you know, that our focus areas are immunotherapy against cancers, and also allergy therapies and other immuno diseases. And we identify clinical unmet needs, and we put them on smart platforms to develop them further. And there is a GI 101 project, and that technological transfer happened in 2019. And we also have joint research partner Yonsei University's Kyungchul Choi, professor at Yonsei University, and we were chosen as top 100 national R&D performances in 2020. And when you visit our company, I would like to show off that we have to create best facilities, best labs, so that researchers can focus on their study subject. That's his vision and philosophy. So we can really show you that we have state-of-the-art technology, devices, and equipment, and our researchers utilize these options and facilities to focus on their research. Myung-Ho Chang, a CEO, was a scientific advisor at Genexine. He was a professor at Osaka University, and he also worked as a principal investigator at IBS, and then worked at Genexine, and then became CSO at Progen, and then decided to start his own company called GI Innovation. And another CEO, as you know, Suyeon Nam, is quite popular. He got MD and PhD at Yonsei University, worked as a medical director at Roche Korea, and then worked at CSO for multiple years at Yuhan Corporation. And then she achieved many milestone breakthrough moments throughout her career, and now she's serving as CEO, CMO, and CBO. Recently, we are preparing to go public, and for that, we decided to hire Junho Hong, who was the first CEO of Interpark Bioconvergence Corporation. We put in a lot of efforts to hire him and convince him to join our team. We also have Yongyu Cho, Vice President at Biologics Incorporation. And there are other team members who are experienced in multiple areas and fields of business and medical. And this is how we are operating our company. Because we are a small-sized venture company, we cannot produce proteins ourselves. So in the very initial stage of our company, we decided to have partnership with CDMOs, and Samsung Biologics have been providing GI 101 protein. They have been very helpful for us, and we're co-developing very smoothly. And for allergen therapeutic, which is GI 301, we are partnered with Binex. And then for GI SmartTab TM platform, We also have antibody targeting technology, and for the adoption of the technology, we worked as investor too, but also working with biologics. And for GI platform technology, which is our representative technologies, we can increase efficacy and we can also bring the cost down. There are smart Clevo and smart selects technologies that we have. And for smart Clego, it is basically encouraging fast cloning by using Lego like block integration. This is why we call the technology Smart C Lego. However, moving on to smart selects, this is what is important is API, and what is important is the bridge role, linker or hinges could be very important because we have to create an stable environment so that proteins can actually become effective, and that is why we decided to call this selects. And as you can see, we have GI 101 and GI 301, and there are other pipelines underway. And for 101, this is being used as a immuno-oncology treatment, and we have transferred this technology to a Chinese corporation, and then currently we are granted with Keynote 101 and now going through clinical stage. For GI 301, we outlicensed to Yuhan, and because it's Japan based, we are currently considering another round of technological transfer to Japanese organizations as well. So we have patents on both GI 101 and GI 301. Because I already covered GI 101, I'm going to be very brief on introducing GI 101. There's FC and there's CD 80 and then FC and then interlunker 2 variants. What is very important is that CTLA4 trap is composed of ECD, endogenous CD80, and the most promising medicine is now sold in the market, but the problem is that it has high side effects, and patients have a hard time to be administered with this product for a long time. So we decided to design a stabler product, and I think this is interest of many other global pharmaceutical companies as well. NK Cells, NKTR signed a global deal with a size of 4 trillion won, and if you look at that specific product, it's failing to increase the temperature up to a specific amount, a point, and we are trying to make it safer by not having to deal with the temperature again. And now going forward to the actual mechanism of GI 101, we it basically interrupts CD80 and CTLA4 interaction with IL-2 variants to activate and proliferate T cells and NK cells. And then as a result, just to be sharing briefly, what matters is that you have to block CTL4 as much as possible. And this is already available in commercially, so we have been checking um, regularly, and you can see that it is encouraging T cell activation by CTR stimulation, and it can re encourage the stimulation very effectively. So maybe there would be a question whether you actually saw this response and reaction, and compared to the conventional RL IL2 CD8 IFN, GL101 has a better efficacy and better reaction, and it also contains a higher volume of cytokines as well. So now going back to the primitive data, when you administer GI 101 twice every 3 weeks, you can see that both T cells and NK cells have been activated. And what is better is that if you think about conventional cytokine, when you do the second dosing, the response drops significantly and then there is a slope. and a slowdown. However, you can see that our magnitude has been retained. Amount of magnitude has been repeated, and whenever you give again and again, the magnitude goes back up to the previous point. And if you think about over here, this essay was created by Professor Choi at Yonsei University. When you actually give a GI 101, we have to see whether TIL, the tumor infiltrating lymphocytes, are going in or not. If you put an isotype control, there's not much being seen, whereas MGI 101, you can see that TIL is being shown, and this means that it is effective when it comes to cancer fighting, cancer treatment. And because we can activate multiple points in a cancer treatment, you can combine other therapies, and we wanted to make sure the results were guaranteed, and we have linked this into clinical study design. So representative data sheets over here is humanized mice. This humanized mice did not have immunity, and then we give a PVMC of human blood, and so that the mice could share the same immunity environment. And then we put breast cancer, TNBC, cancer cells in order to carry out experiments, and we give Cambridge Marketuda at the same time, and then you can see that when we put GI 101 alone without the Kembruda, the effect was good enough. And when you did combined therapy with Keytruda, you can see that the efficacy went up as well. And it also showed that it has a memory feature as well at the end. And VEGF inhibitor or reduced therapy could be combined with GI 101, and it showed a synergetic effect for sure. So, with utilizing these results, we are now currently designing our clinical studies, and we were granted with PKOT, and we are going to carry out joint research with them. And as I said, we did technological transfer to Simcere. It's one of the Chinese innovative companies approved by the Chinese government, and about 900 billion won size of a joint technological transfer was carried out. And by utilizing the clinical results in the future, I think the transfer growth is going to be expected. And another type of technological transfer was related to Yuhan, so we did not do separate IR for this. And for Japan, it is our strategy to exclude Japan because we have to carry out a specific clinical study separately. And it seems that their regional patient group is different from other countries, so we're going to we carried out a technological transfer with a different approach, and joint clinical studies are going to be carried out smoothly, I believe. Sorry, I showed this wanted to show this earlier, but couldn't because there was some screen error. Now I have to talk about next pipelines. Now after GI 101, we have other pipelines underway. For there's N series, there's the 2 series, and there's 3 disease, which is about metabolic diseases. And for GI 10N series, not only CD8 T cells go up, but other NK cells are successfully increased in number. We are going to be specifically targeting NK cells by using GI 10N, and these types of medicines, we always have to think about side effects. For example, this is also the monkey data, and you can see the body mass and weight has been decreasing consistently, but our medicine, it doesn't show any weight loss, and it is indirectly proving that it's safe enough. And then another data that is presented is global pharmaceutical companies are developing NK cell activators and T cell activators. But compared to their open and public clinical data, I think our NK cell activator is going to be better. Because this is monkey data, I believe that this results can be successfully transferred to human bodies as well. Thank you very much for your attention.
Thank you very much. We'd like to also thank to Young Jun Ko, the head of Discovery Division of GI Innovation, for his informative presentation. Also, please have a lot of attention and support to GI Innovation, which is leading the development of biotech for patients around the world. Next, there will be a presentation by Jun-Heng Lee, CEO of Baxel Bio. So he's now on the stage. Please welcome him with a round of applause.
예, 반갑습니다. 저는 백신을
Good to
두 번을
see you
다
all.
맞았거든요. 근데 이게
And I got vaccinated twice, and when I keep my mask on, I just I have issues with my glasses, so please understand. Vaxel Bio is a company which is more than 10 years old, and as a co-founder, I work with Professor Lee Jae-chung, and who entered the same medical college to my a few years later. And I do hold MD degree, but I do not cover patients, but I engage myself in the research and work on biotherapeutics. And we founded this company together. And the key strength that of Vaxilla Bio is that I focus on basic medicine and mouse samples. And my co-founder focuses on human patients. So the strength is that unlike other startup companies that have difficulty entering the hospital market, but we have this key advantage here. And as CEO Zhang mentioned, when engaging in this research for over 3 decades, whenever there is any challenge, that I do get a lot of help. And basically, our company, Vaxel Bio, other former companies showed preclinical data, but our company, our focus is that under the given circumstances, our key strength is in that era. So we focus on clinical phases, and we have business developing strategy in that too. And we have 10 mottos, and we remind ourselves every time we have a business meeting, and one is that make our patients alive. So our goal is to keep our patients alive. Vaxel Bio, based on this, when we worked on IPO, we got this skeptical question. So when in terms of immune mechanism and based on immune mechanism, when we try to kill tumor cells, if we purely concentrate on one part, it doesn't work. So that is what we learned over the past 30 years. And I often make a joke when if one person says I'm going to overcome cancer with one single treatment, then it's either this person doesn't have any background knowledge, or either this person is a scammer. So in order to develop a cancer treatment, we consider all the multi-factors, and we take a multidisciplinary approach to come up with a pipeline to effectively treat our patients. And from innate immunity to adaptive immunity, we cover all of these 3 ideas based on our pipeline. And when we first began our clinical study, is in order to connect innate immunity and adaptive immunity based on our platform. And similarly, this may sound primitive, but just like GI innovation, we use cytokine as part of our pipeline. So ultimately, these all attack tumor cells, and we have to keep our patients alive, and that is our key. So first of all, the pipeline that we have, we just completed our phase 1 and we're working on our phase 2 clinical trial. And our goal as an NK therapy is that we kept advanced carcinoma. And if you put it in simple language, it's just terminal cancer. So those patients have already received a lot of other cancer treatments and they came to us. And we, in terms of human microenvironment, we just physiologically, if we let this cancer cell let it go, then we don't know what will happen. But if we abruptly change this, and if we inject NK cell into that room, then NK cell will effectively kill tumor cells. So our strategy here is that HAIC, which is hepatic arterial infusion chemotherapy, and we inject this. We inject this tube and inject this drug, and itself will kill tumors and it will suppress tumor and it will change the environment. And we find this vacant room and we use this chemotherapy. And we extract blood from patients, and within 2 weeks, we inject this. And compared to other peers, we conducted research, and the dose of our administration is 5-fold or 10-fold, and it is effective. And the way to make people survive is that we have to secure this pure technology, and we have to secure safety. So we increase our dose to increase the efficacy, and that is our strategy. And so when we gave it a shot, for those terminal carcinoma patients, and there is medicine called phenomena, a terminology. So it can prolong patient's life for 3.3 months, and worldwide this has been tested, and it is written on Wikipedia. But this can only extend life for 3.3 months, but the response rate is only 11%, which means only 1 out of 10 patients can survive for 3 more months, so only 10%. But when we adopted this technology, the response rate increased to about 73%, and the efficacy was about 81%. So, and another noteworthy characteristics is that the life prolonging characteristics. Of course, we did expect some bright efficacy, but this was more than we expected. And January was the last time that we did follow-up research. And based on that research, we could prolong patients' life for 40 months. So our patients are still alive. So in real-world data, is probably even better than this. So, and also, if you take a look at the left, there is no severe side effects at all so far. And here is an example. If you see these arrows, and you can see that cancer reoccurred. But after our treatment, when we took a picture, the tumor disappeared, and the markers dramatically decreased. And our patients, there are patients who decreased the tumor size even more. So I show you these two pictures to show the difference of tumor size. And we began our second phase, and the second phase clinical trial is slightly different from the first one. And we have our know-how and our technology, and we increased our technology and expanded our knowledge. And when we extract cells, especially from cancer patients, and if we freeze that and melt it, and if we try to administer that, then usually the activity, more than 10%, is gone. And like 20%, is a very important factor. So we have to do our best to keep the efficacy. So and we built our know-how. And compared to phase one, we decided to increase it, and we administer this agent for 5 times, and within 1 month of period, we got approval and we began our clinical trial. And when we changed this protocol, we got approval from KFDA, and we select patients and we increased the dose twice and conducted the evaluation study. And if they approve us and tell us, then we can move on to the next stage, and that's the processes have been done. And next week, we will move on to SRM, and we will notify this information in the next stage. And when we got approval for 2 groups, and thanks to our good news, there are 4 more hospitals that wanted to adopt our technology. So I guess it'll propel the speed. And next is DC. And Professor Lee has been conducting this research for very long. And when we first conducted clinical trial, then this would be the research. So our DC is a very strong one, and we did confirm that efficacy. And then we conducted clinical trial in this way. And Professor Lee, I would say, has secured the most number of patients of this, and we did got a treatment, and we just completed 2A clinical trial, and we're moving on to 2B clinical trial. And next one is another thing, which is CAR treatment. And we are working on developing a CAR treatment. And of course, among our developments, there are CARs for multiple myeloma, for example, CAR T. But in fact, that field, from my perspective, there's Professor Kwon, but in terms of hematology treatment, Curacell or other companies are already spearheading in that field. So it's a very bloody red ocean. So we would rather concentrate on developing solid tumor treatment. So there are about 4 CAR T developments, and we are currently conducting animal experiments, and we're conducting various tests. So I'll just briefly introduce one here. So compared to hematology cancer, a solid tumor, there is high level of antigen escape. So if single histronic CAR is used, then the efficacy is really low, according to various research. So if you combine different CARs or work on multi-system CAR, then if you try to do so, and this SCFB size, actually, if you inject this into a cell, then it doesn't fit well. The bigger it is, the more difficult it is to inject in a cell. So we came up with this monobody system, which is 1/3 of the size of the conventional one, and we conducted this on animals. And when you use this, you can come up with multi-cistronic construct, and you can target this carcinogen, and you can overcome various conventional challenges thanks to this CAR T technology, where you can increase the efficacy. And this is our cash cow, Bio-15, as you know, can increase NK cells and proliferate it with cytokine. So we developed cancer treatment for puppies, and nowadays cancer treatment market for animals is significantly increasing, and compared to people, dogs get a lot of cancer. There's some if you do the math, dogs may contract cancer 35 folds with more chance compared to human beings, and if they get older, even 50 times. So we conducted clinical trial and currently waiting for approval. and our test's result shows that the satisfaction rate of animal owners, dog owners, is almost 100%. And there are exceptions, is that there were patients who completely treated their dogs thanks to our treatment and they didn't come back, so we couldn't ask their satisfaction. And as I mentioned, doctor, the way that autonomous survives is that we use protophase medicine and we have to stick with the principal technology. And automation, smart factoring is the answer. So at first we would say Robert Schultz, when he founded Starbucks, and when he said, "I'm going to sell coffee to the all around the world," and everyone laughed, "Ah, you're crazy." But nowadays, because of Starbucks, all the other famous coffee companies are just closed their business. So how you work on expanding your business changes the way you can survive. So we will build this smart factory based on a certain level of completeness, and I would suppose that this market won't be that difficult. So we will keep working on being autonomous. Thank you for listening.
Thank you very much for your impressive presentation. I also could find that Evoxel Bio can provide the promising future to patients by supplying a novel therapeutic options. Next, there will be a presentation by Kon-Soo Kim, CEO of Curacell. So he's on the stage. Please welcome him with a big hand.
Good morning, ladies and gentlemen. My name is Kom-Soo Kim, CEO of Curacell. As you know, that we are a developer of CAR T immunotherapy, and maybe you have already listened to this, but it is considered a dream cancer therapy or cancer therapy of hope. And you might wonder why this is the case. And I'm going to show you and explain the principle of CAR T therapy and what we're currently developing. If you look at this image, this is the imagery of terminal cancer patient. These patients usually have already used all these options and they do not have any more options left. If you look at the images, all those cancers are dominating the body on the left image, and then within 1 month of the administration of our treatment, it's almost gone, and treated with the cancer. And there are two approved CAR T options. One is CD19, and there are four different products: Kimli 19, and Naticartas, and then Brianzi, and then Yescarta. And then early this year, BCMA CAR T was approved as the second type CAR T in the world, and this is called Ave CAR T as well. So using single medicine and being able to treat a cancer, this is called single therapeutics, and it is basically used as a single dose administration in order to remove almost all cancer cells in a terminal cancer patient's body. It takes about 10 years to come up with a new development. That's true. That's common. However, if you think about Yescarta's development, after pre-inhibiting to the actual approval, it only took us 6 months. Why is it possible? It's because single dose administration, and within 3 months, you can check and identify the reaction of patients' bodies. And if you think about early this year, in Korea, CAR T clinical studies were not going on at all. In China and in the US, there were more than 300 CAR T clinical trials. So Korea was excluded from the scene, but for the first time in Korea, we received the approved approval, and as a result, we're now going through the early stage of CAR T clinical trial. Now, let me go to the main principle of CAR T therapy. On the left, there is natural T cell reactor, and you have to understand how T cell is basically killing cancer cells. It is simplified, but there are many different molecules that are involved. There's MHC TCR complex that is recognized by T cell, and then once it is judging that it is a cancer cell, then it does immune attack, and this complicated mechanism has to evolve many enzymes and reactions, and CAR T is basically simplifying the process. What it does is that it puts in the order of antibodies, and then it activates tumor cell T cells by utilizing other molecules, and that's called CAR T therapy. The concept itself was developed a long time ago, which is the 1980s in Israel, and then after going through many different rounds of development, and now we call next generation CAR T when CAR T technology includes other molecules. Currently, commercially available and widely used CAR T therapy would be second generation. And the simple mechanism is that there's a single chain FB will recognize a specific antigen, and the signal that's recognized is going to activate the CDT ejecta or co-stimulation domain in the cell, and it will be amplified. And then the sleeping CAR T cell will be awakened, and then that'll activate a T cell, and then it'll proliferate, and then as a result, will remove and eliminate cancer cells. By going through that phase and steps, cancer will be treated. And now let me discuss how cancer is fought by CAR T. What's most important with the partnership with hospitals? What we used to do is basically pharmaceutical companies send and ship medicines, and hospitals simply use them. But CAR T therapy is different. During the manufacturing system, the hospitals intervene and put impact. So basically, what you do is we do pre-treatment testing when we create and collect lymphocytes. So it's basically in the hospital. There is a dialysis medicine, a device. It's not exactly dialysis device. It's similar to that. So utilizing that device, we collect lymphocytes, and then we send this to GMP of pharmaceutical companies, and then by utilizing that sources, we culture T cells, and utilizing different methods, we will introduce other genes, which is there could be a non-viral vector option or viral vector option. And by utilizing a massive culture method, we achieve the target dose for a specific patient. And after that, what they do is the factories freeze the manufactured CAR T, and then the cancer patients will go through lymphodepletion. In order for the CAR T therapy to be implanted in the human body, lymphodepletion process is very important. Patients' lymphodepletion has to be done before CAR T to be injected. That is why CAR T has to wait in the freezer. And once the lymphodepletion is done, then the CAR T therapy medicine will be shipped to the hospital, will be thawed, and then put it into a patient's body. This is only for one patient. So think about costs, ingredients, and labor costs for this one single patient. That is included in the cost of the medicine. As a result, if you look at the right side of the image, the overall cost for CAR T therapy, the cheapest would be like 500,000 USD, which is 500 million won. And if there is side effects that are appearing in the body, then maybe the maximum will be a billion won. And there are many CAR T therapies that are approved, and it showed great results. But there are homework and hurdles that CAR T therapy has to overcome. First would be even the patients that completely removed and eliminated cancer, cells in 1 or 2 years, it can sometimes come back. So we have to think about how we're going to handle long-term survival of these patients. And secondly, if you think about lymphomas treatment results are different. So lymphoma has a twice better result than cancer. So it depends on the types of cancer. Depending on the type of cancer, the treatment rate differs. So we have to understand how we're going to increase the treatment rate, the completion rate, depending on the types of cancer. And now we have to think about how we're going to more effectively target solid cancer rather than lipid cancer, because solid cancer CAR T therapy has a very, very low rate of success. Especially PD-1 blockade antibody, if you put them together with CAR T, we can increase the efficacy by a little bit. There are some reports about it. And in our company, we call a new technology called OVIS. And now let me go into detail about OVIS. So now you have to think about why CAR T is not effective for solid cancer. There are many different explanations, and this image has some content. So what we need to focus is that there are immune cells in the tumor, and then because they interact with cancer cells for such a long time, PDR immune strengtheners representation is already heightened, and that is why CAR T therapy is not as effective. As a result, PD-1 antibody is now jointly used with CAR T therapy in order to overcome that challenge. And what we're trying to do is how we're going to inhibit the immune strengthener. And as you can see from the left image, we represent CAR, but we also make sure that PD-1 and TIGIT to be inhibited. So theoretically speaking, CAR is right there, the CAR technology is right there, and then we also use inhibitor of PD-1 and TIGIT. So it's a combination of these two technologies. So when PD-1 and TIGIT are there, of course, CAR will activate T cell activation. But as you can see, because PD-1 and TIGIT were already removed by our technology, CAR T can more successfully and efficiently target cancer cells. So we carried out animal clinics, and then as you can see, if you compare conventional CAR T and our Ovis TM CAR T, depending on doses, there's high dose, medium dose, and low dose. And this is also lymphoma cancer cells. And we also use T cells of a healthy person. And for high dose, both showed great efficacy in treating cancer. But if you go down to low dose and medium dose, you can see that Ovis TM CAR T is showing better efficacy than conventional CAR T. And now next one is the pre-OC data of Ovis CAR T. We also divided them into 3 different doses. And if you compare conventional versus Ovis CAR T, and you might not be able to see the picture very well, but if you look at the 3 graphs, for the tumor version, Ovis CAR T showed a significant drop in the graph. And the blood T cell were blood cancer patients' T cell. And if you go into Ovis technology, there are many different attempts and trials in overseas as well. You know, Caribou Biosciences, which is famous for receiving Nobel Prize, they are using CD19. It only specifically removes CD19 receptor inhibition. But now, instead of that, we are putting PD-1 and TIGIT neutralization at the same time. We already proved through our basic research that removing PD-1 and TGIT at the same time can show and strengthen the results. So we received healthy blood from SNU hospital, and then we have created 100 batches already, and then we also received blood cancer patients' blood, and then we created 25 batches, and based on the results of the study, we have received the IND approval. Now moving on to the clinical trial design for OVIS CD19 CAR T. Our first goal is to check safety, and then we also will check efficacy as well. It is 3+3 design. We're going to do a low dose, or medium dose, and then high dose. And then you can see that three patients that belong to cohort 1, 2, 3 is going to go through the increasement of dose administration for their bodies. And we have a very close partnership with Samsung Seoul Hospital. We will utilize GMP facilities in some Samsung Seoul Hospital. About 80 to 100 patients CAR T doses can be generated and created. Currently, we have a POC clinical trials for CD19, but we also would like to go into TPD, lymphoma, AML pipelines in the near future. And eventually, for solid cancer treatment, we would like to develop first-in-class CAR T therapy program. This is our mid and long-term goal. Thank you very much. But because of time constraint, would you please conclude your presentation?
Thank you very much for your cooperation. Since we have a time limit, please keep the guideline of the time that we have informed. Right then, we will move on to the next company presentation. There will be a presentation by Seok-eun Nam, the CEO of Neona Therapeutics. So please welcome him with a round of applause.
Thank you. Good morning. I am Seok-eun Nam, the professor. Actually, I'm a CEO of the company here, the Neona, and also currently I'm a professor at the Department of Pathology, College of Medicine, the Catholic University of Korea. Actually, I was informed that the official language is the English for the presentation. But, you know, I saw all the speakers give a presentation, our language Korean. But I already prepared the presentation by using English, so if you don't mind, I give my presentation by English, okay? I think this presentation is the very little bit a first step toward for our future collaboration or investor relationship between you and our company, Neona. Okay, I don't think many people know about my company, Neona, because this company has been established in last August, year 2020. So it's a pretty new venture company. It's been less than 1 year. Actually, like most of venture company located in the university campus, our Neona is also is the research-based and the anti-cancer drug development company based on my more than 20 years of the research career in RNA cancer genomics field. Okay, this slide shows why we named the company name as Neona. As you know, neo means the new things, and we pursue the new concept of the RNA therapeutics in especially cancer. So we put these two words into the single name as the Neona. So our company Neona develops the RNA and oligonucleotide therapeutics that target RNA and its regulatory factors in the processing of the RNA networks. Actually, the RNA life cycle is very, very complicated, as you can see here. The RNAs are regulated through the dynamic and diverse process within the cells, and also these complicated RNA regulatory networks are regulated by various factors, which we call them RNA regulatory factors. So our company, Neona, aims to become a leading company in the development of RNA therapeutics that selectively target RNA or RNA network regulatory mechanism for disease especially for cancer, based on our original technology for clinical naval verification research data. Our company located in the Seoul St. Mary Hospital campus, as you can see here, just next to the hospital. Here is the medical school building. We are here, and also you can see some forest area which is heavily under the construction for new building complex. It will be finished at the end of this year, and we are planning to move this area in next year with some space area. When we start our company, we prepared 4 different pipelines. Of these 5 3 pipelines are basically the RNA targeted and RNA-based anti-cancer therapy. And the last one is the multi-panel-based, the early diagnostic the tools, especially for the liver cancer. Of these, the 3 RNA-targeted RNA therapy, the 5 lines, the first one is especially focused on the small microRNA, LES7I5P. This LES7I5P has a very pivotal role in the regulation of macrophagy. And then the second one is we recently found some RNA splicing factors are deregulated in the development of the liver cancer. And also, we found some enhancer molecules are also deregulated in the development of liver cancer. So we targeted these molecules by using siRNA techniques. Today, due to the time limitation, I'm going to show some data about the first 5 line, which modulate macrophage immunoactivity to cure the cancer. In normal physiology, macrophages recognize the damaged cells to clear them, and at the same time, to avoid hyperactivity of the macrophagy, most of normal cells interact the macrophage cells by interacting with the specific ligand sulfur alpha molecule with their specific receptor CD47. However, recently many scientists discovered many different kind of cancer cells mimics like these normal cells. They do express CD47 molecule and also interact sulfur alpha ligand to pretend as a normal cells, and then by using this they can avoid the macrophage attack. So many of big pharmaceutical companies now, they are developing CD47 antibody, which interfere the association between macrophagy with the CD47 tumor cells. However, unlike this mechanism, our company discovered a very unique mechanism which modulate the immunity of the macrophage cells. In liver cancer, histone deacylase 6 is a very potent tumor suppressor gene. And this HDAC6 should suppress LES7I5P in normal status. However, once inactivated or suppressed, these LES7I5P microRNAs are re-expressed so that it can suppress the thrombospondin molecule. This is very important molecules that act as a endogenous anti-angiogenic molecule inside of cells, so that it cannot interact with CD47 receptor. So this CD7 receptor is open to bind to the sulfur alpha ligand of the macrophage. So this switch the status of the macrophage as the emit signal to don't-emit signal, so that macrophage no longer attack tumor cells. Here is some data. we harvest mouse peritoneal macrophage and co-incubate with human tumor cell SNU POD23 cells. And at the same time, we treated it HDAC6 plasmid or antisense oligonucleotide of the LES7I5P or the thrombospondin proteins. And as you can see here, all these 4 treatment enhanced macrophage phagocytosis activity. As you can see here, the fluorescence color means macrophage already eat the tumor cells. And when we pre-treated 3F352 antibody, which is the anti-CD47 antibody that specifically blocks the binding of thrombospondin to CD47, they are, as you can see here, dramatically inhibit all these 4 different treatments. So with this result, we were able to make a conclusion that, as I mentioned earlier, in normal status, HDAC6 should suppress LES7I5P. However, once inactivated or suppressed, it express it to inhibit thrombospondin proteins. Then this causes to open the CD47 receptor so that binding of the sulfur-alpha ligand to the cancer cells. And then, as I mentioned earlier, this critically switched macrophage to the don't emit signal status. Okay, here is some in vivo data. So we prepared the animal model, which developed the endogenous liver cancer around at 20 weeks of age. As you can see, the heavy liver mass and numerous liver cancer mass in the animal model. However, when we treated it, HDAC6 plasmid or antisense LES7I5P or thrombospondin, all these treatment significantly suppress the instance of liver mass or size of liver mass in the same animal. And the immunohistochemistry, we were able to confirm these results by observing as you can see here, the staining with specific macrophage marker F480 staining shows that the lot of number of macrophage compared to non-treatment. So our pipeline, the LES7 therapeutics specifically focus on the LES7 I52 micro RNA so that it re-express the thrombospondin protein, then it can occupy the CD47 receptor, and then this interfere the association between cancer cells and macrophagy for the don't emit signaling and to the switch it to the emit signaling. Okay, currently, as I mentioned earlier slide, many of big pharmaceutical companies, they're developing anti-CD47 monoclonal antibody, especially for the non-Hodgkin's lymphoma or ovarian cancer or other type of cancer here. Because all these cancers express relatively high level of CD47, molecules. Then what happened to the other cancer? For example, like liver cancer, as you can see here, they show the relatively low level of CD47 expression. And more, when you look at the CD47 differential expression in various cancer, you can only find the 3 cancers shows differential expression of CD47 molecules in cancer compared to corresponding normal. However, okay, skip this one. However, when you look at the differential expression level of thrombospondin molecule in various cancer, we can find more than 60% cancers. They shows the very suppressed expression of this thrombospondin molecule. So our approach is to regulate the microRNA LES7IP strategy can be applied to broad range of the cancer. All these data has already published in the hepatology, and we hope we can enter the phase 1 clinical trial in year 2025. So we are currently spending very hard time to develop these therapeutics. Okay, I think I must stop here, and I appreciate for your attention. Okay, thank you.
Thank you very much for your informative remarks, Professor and CEO Seok Eun-nam of Neonat Therapeutics. So far on the topic of new therapeutic strategies for NK cell targeted immuno-oncology at session 3. We have listened to the topic presentation and discussion and company presentations. From now, we will have a time for a break, and then session 4 will continue from 1:30. So the online participants will also take a lunch break, and then let's meet again in session 4 at 1:30. Thank you very much. We'll see you soon. Ladies and gentlemen, may I have your attention, please? In a moment, session 4 will start at 1:30. We would like to thank all the guests for entering the venue and be seated. And also simultaneous Korean English interpretation will be provided for the event. And channel 1 is for Korean, channel 2 is for English. And you can download company briefing material files through official website. Welcome back to Bio Korea Invest Fair 2021. Did you have a good lunch break? Hope you did. Session 4 will proceed with the topic of bispecific antibody platforms for next-generation cancer therapy. So firstly, there will be the topic presentation for Session 4. We will have a Sang-Hoon Lee, PhD, CEO of ABL Bio, onto the stage who will give a presentation with the topic of bispecific antibody platforms for next-generation cancer therapy. So please welcome him with a round of applause.
So today, before I summarize some of the bispecific antibody trend in cancer, the program, I want to highlight, the first that, as the older program for any new drug development at this moment has a lot of time and effort and finance, the researches such as about, the 2.5 billion US dollar cost more than 10 to 12 years, and then including, the 1.4 billion US dollar about out-of-pocket cost, and time cost is about estimated 1.1 billion. So in contrast, about 10 years ago, the Tufts University estimate about the 800 million cost for new drug development, but now it's more than double, the 145-fold increase. I think not only the biotech but also the large pharmaceutical company, most the R&D has an issue that the productivity is not as strong or fast as the long time ago. Internal resources, for example, during the COVID-19, I was told by my so many the US colleague previously that they didn't actually go to the work. So in other words, they shut down a lot of R&D activity. However, as you see so many reports, Big Pharma actually continuously do the M&A, the license deal continuously. So I think they have a certain budget, but they have some projection to do the some R&D activity. So now they are looking for outside the resources, for example, licensing, M&A, etc. I'm going to talk about a little bit later. I think the big pharma and biotech is such a good example for the open innovation. For example, pharmaceutical company has a financial resources and manpower, and then commercialization activity. And biotech usually have the discovery engine and then new technology. So I think both the sector can merge together and marry together. Then the lead open innovation. But I think the key factor for this even open innovation in Korea, the open innovation is one of the famous the terminology that every single company utilize. We're going to do open innovation, but I think that it's basically science-driven decision. And no matter what, I think either preclinically or clinically, the big pharma see the data from biotech or technology that they decide to apply to the open innovation area. And I think the 5 different areas I'm going to talk about, from left to right, you can see that the scientific the expertise. So in other words, either technology-wise or science-wise, it should be good. It should be great enough for the big pharma to pay attention. And I think the differentiation of the pipeline is a very interesting one. Either the molecule should be the superior or show the differentiation. So I think that's another one. In the middle, I put the innovate platform. I think I'm going to emphasize a lot of the key factors for platform strengths, because I think having platform can expand the pipeline, not only one pipeline, but also multiple pipeline continuously. I'm going to touch a little bit about ADC later, but in antibody drug conjugation area, Legochem is the key leader in South Korea, but ADC field has been struggle last 30 years, but last the couple years ago, now ADC renaissance. I mean, the third generation ADC came out, the technology become much better, and faster movement. I mean, we know how to tackle the technology. And now right part is the integration capability, revenue generation. I think in South Korea biotech, it's very hard to imagine revenue generation at this moment because revenue generation coming from commercialization or royalty. But I think none of the biotech yet or pharmaceutical company yet to come to global commercialization yet or global loyalty continuously. I'm going to touch base a little bit about the Morphosis case, in next couple slides. And key driver, I don't have to mention about the one by one, but I mean we should have some very strong pipeline. Corporate performance, including the human resources, we should have a good the manpower, and then good science. And again, I mean we should have a good partnership. So the partnership is a good marriage. So you can imagine in marriage, the wife and the husband initially they work together, but we fight each other. We come back together. So I think the between collaboration is very similar, the pattern, the both companies should have a collaboration spirit, and platform differentiation is one of the key factors in my opinion. In the oncology area, I'm going to touch base a little bit. So key factors has been around, the like angiogenesis, like Avastin or small molecule sutent. I mean, touch base, the vessel formation inhibition. So it's been, the long history that, if we inhibit the angiogenesis or stroma area, tumor microenvironment, we can inhibit the tumor growth inhibition. And I think the ADC technology definitely third generation is the emerging antibody drug, the conjugate. And as you imagine, this one is the target therapy, like, the Avastin, Herceptin, Erbitux, etc. We basically target the either antibody or small molecule, then, inhibit the pathway of the tumor pathway. Last, the 10 years, definitely immuno-oncology area is one of the key area, not only vaccine, but also CAR-T, and now today, bispecific antibody, I'm going to talk about it. In an antibody field, I think usually it's a monoclonal antibody, everybody knows. And then there's the emerging technology, ADC. Now, used to be IV injection of the antibody, but, even Korea, the Alteogen and other company has a sub-Q injection. It's more convenient way to deliver the drug. And I think the glycoengineering is one of the technology. And now we have some multiple bispecific antibody, either two target antibody or one antibody bind to the tumor target, the other one bind to the T cell. So we have a T cell engagement. Bispecific antibody, the growing emerging, the area. You can imagine that about within 10 years, we can have about more than 10 billion, the estimation. It's a fast growing area. When I talked to one of the colleague in Genentech, he said, I mean, even Genentech and Roche pipeline, especially for oncology, large molecule antibody area, 85% are right now bispecific antibody. So there are definitely a lot of bispecific antibody are fulfilled in Genentech Roche pipeline too. And this diagram is a very simple area. So definitely you can have an antibody base. You also have a non-antibody base, the technology. But one of the famous bispecific antibody called BITE, the Amgen acquired technology long time ago, single chain FB, small fragment, conjugate. Although it has a very, the short half-life, Amgen has also long half-life extended. And now they are doing a lot of clinical trial. I have one slide in, the later. And we have, the multiple, the bispecific antibody. In bispecific antibody, more than 80 different formation of the bispecific antibody are existing. But I just highlight the circle that these kind of formation are the very popular, the famous, largely, the used formation. For example, Genentech Roche, Napinol technology, and Roche, the cross-map. And I think that this one is the EPI, DVD. And this formation is the Morrison body, has one, the large molecule IgG, and then single-gen FB, etc. So it all depends on, the company. everybody claim they have a differentiation. So either the differentiation should come, either manufacturability, efficacy, and the now side effect, etc. I'm gonna skip this slide because bispecific antibody can use a lot of time, the immuno-oncology and CNS. You can see that the big pharma has a lot of bispecific antibody the field, and the relatively large size between 1 to 5 billion market size. Famous company called Genco are one of the leader. And then you see the molecular partners, the Canadian company Zymox, and the US company Macrogenics lead the medium market, and then a lot of small market size company including Meros and Eduro and Sutro and Merrimack, etc. So I think you can see that the more than the 30 different company are developing so many different formation of the bispecific antibody. And I think that I want to highlight that the last the 5 years, especially from 2017 2019, the multiple deal came out between large pharma, Merck, and Amgen, the Seattle Genetics and Genentech and GSK, etc. And there are the cases, the either platform plus the multiple formation of antibody or 3 different target, etc. The contract is the relatively large. So in other words, big pharma used to buy as a proof of concept 1 molecule plus apply their technology. Therefore, they continue collaboration project to generate the 3 to 5 different the early stage molecule. And I think that I want to just show the slide, just showing the formation. You can see that this kind of formation of bispecific antibody, Amgen BiTE, and the like one arm to the other one, napinol. And the other formation is there are multiple formation, the diabody, etc. The definitely, the show different formation. And I think the even, the same 1+1, there are the different, the differentiation came out by many different company. And I want to highlight one interesting company, the company called Genentech, definitely bispecific antibody pioneer or antibody area pioneer. And initially they started bispecific antibody, the 1+1, napinol. So like target A, there's definitely 2-arm, 1 antibody, and then target B, 2, the 2-arm, the other antibody, and they cut down 1/2, and this cut down 1/2, and then conjugate together, called the nabintol. So this is the typical, the first generation of the bispecific antibody Genentech decided to have. Now, for example, Genentech, you apply their napinol to CD3 bispecific antibody. For example, they apply to Herceptin here and CD3. And while they do the preclinical study, also clinical trial, they realize that having this form, there are high affinity CD3. Therefore, they actually modify CD3 to low affinity to minimize the side effect. But to improve the efficacy, they interestingly generate the 2 binding site of Herceptin together. This is basically 2+1, the asymmetric formation of the bispecific antibody. So I think that this is a very interesting concept, how they evolve from 1+2 to 1+2. This year, AACR is surprisingly the 2nd generation T cell. Since ABL Bio is more focusing on 4-1BB, I want to have the slide summary 4-1BB. But in my opinion, 1st generation, the T cell engager is the CD3 bispecific antibody, either BiTE, the lead by Amgen, or the Pfizer, Genentech, Regeneron, etc. But however, in this, the year AACR, more than 16, the 4-1BB antibody field, that were published, including ABL Bio. You can see that there are so many different formation of the 4-1BB. Why 4-1BB? 4-1BB, the next generation of the T cell engager, is relatively much weaker than CD3, but it has a memory T cell activation. So it's an emerging target and emerging technology in this bispecific antibody. And a lot of companies are preclinical, but most leading companies are just in the phase 1. So a couple companies just finished the phase 1 and ready to move to phase 2. And I'm going to switch the gear, the showing multiple company cases utilizing bispecific antibody. The Genco is one of the key cases. They have so many different pipeline with so many different formation, either 1+1 or 1+2 asymmetric, etc. And they also have a multiple, the partnership. For example, either, the small, very similar size company Morpholsis and larger size, for example, Boehringer Ingelheim, Janssen, and Merck, etc. And also they have an internal pipeline, the development. I'm going to touch base, a little bit, yeah. And Morpholsis is a very interesting technology that they have a lot of the pipeline. But recently, Morpholsis acquired the company Constellation Pharmaceutical Company. This one is the totally different the modality company. How they do that? They actually build a platform. They have a partnership, out license, and revenue generation. But finally, one of the product generate a huge number of the revenue just because they had a royalty. That with the royalty, with the cash, they were able to, to generate the M&A deal. And I'm going to show the Regeneron is a very another interesting company. Since I don't have enough time, I'm going to skip the just showing. And the Byte is a very interesting technology also. And Seattle Genetics is used to be ADC technology company. Now they also have generating a lot of revenue by 3 different products. Now they generate the royalty, also the internal product, the commercialization. Now Seattle Genetics, we don't no longer call the ADC company. It's the pharmaceutical company. They have a ADC plus immuno-oncology program. And I think the immunomedics is a very interesting one. They develop ADC. Finally, Gilead actually acquired the immunomedics with a 21 billion market size. So I'm going to highlight, finally, in Korea, I think at least since I'm presenting to Korean audience, so Alteogen is a very interesting platform, the sub-Q injection technology. And Legochem Bios, the definitely had a great, the conjugation technology for ADC. And Hanmi has, the lab discovery program. And ABL Bio has the bispecific antibody. So I think the platform, I'm going to talk with, the CEO Mangsun Hwang about, about the field and, some trend later. But I think at the final slide, not only I'm showing the bispecific antibody field, but also generally in the oncology drug development, we do have a lot of the open innovation to improve the productivity and reduce the failure. So you can imagine the benefit of partnership is one of the key factors for the success of the small biotech working together, larger biotech, the biotech or pharmaceutical company. I think that I want to stop here, and then we'll continue discussion with Mr. Hwang. Thank you. Yeah.
Thank you for your impressive presentation. So it was the presentation on bispecific antibody platforms for next generation cancer therapy. Now we'd like to proceed with a discussion session. So as debaters, I'd like to introduce Sang-Hoon Lee, PhD, CEO of ABL Bio, and Mansun Hwang, CEO of Korea Investment Partners. So we have 2 gentlemen on the stage. So please proceed with the discussion.
So I'm informed that we can conduct this discussion session in Korean. So when I have listened to Dr. Lee's presentation dozens of times, and every time I think you were a really good presenter, and you delivered a lot of dense information. Of course, there were some ideas that you skipped, but all of the ideas closely came to my mind. And today, there wasn't a lot to talk about ABL Bio, but in investment or biochemical industry, I have a couple of questions to ask. Among the ideas that you delivered, the platform technology, you mentioned this as a key idea. And in terms of platform technology, does it have expandability, and does it have meanings? And since we are a company located in Korea, and when I interact with other overseas company, and all the companies in the past said that they have their own unique companies, and nowadays all companies say that they have their own platform. But is it really that all the companies have their own platform? If a company wants to say that that company actually has a platform, what kind of characteristics does that company have to have? And if you have some kind of technology, does that can that be called a platform and POC? To what extent do you have to have? And IRGO Biden or LBL Bio, among other companies. When a company has a platform, what kind of characteristics do you have to have?
Well, as I was listening to your answer, I think I can categorize my answer into two. For example, let's say there's a domestic company that has the platform technology and also a US company that has platform technology. If you compare the two different categories, let me go first to the US, MorCosis, the Seattle Genetics, Amgen Genetic, Regeneron. These companies used to have platforms. So they had platforms, and then they went through different failures, and they also went ahead with the clinical trials. And then Seattle Genetics decided to come up with the next generation technology. So one technology could treat many different applications, but I think let's say one therapy, if it is proved by one disease, one application, then I think that platform it has a scalability. However, if you think about the Korean side, let's say the companies, including myself, let's say we just hit the clinical stage, then we are now at a relatively premature stage compared to the US. We are still at a baby step. However, Korean platform companies or biotech companies, we designate a POC material, we do IND, and then we do clinic. Let's say we prove our results, then there is a chance for validation for that platform. So we can judge that based on the result.
Thank you for your answer. And you, including ABL Bio, for companies, if they take timely measures and show that they actually have platforms, and if they become secure their positions, then it will be really meaningful. And also, another question is about small molecule. So there are cases when they fail developing, and sometimes they fail because of adverse effects. And when we talk about new drugs, then there needs some efficacy. And also, people say production is the key. And when you develop a new drug, I haven't actually heard that production is the key. But for biopharmaceutical companies, you have been engaging in this field and developing new drugs. And in terms of new bio drugs, in terms of efficacy or development, any ideas?
I'm sure everyone or many of you know, but there's small molecule, large molecule, and the difference between antibody. I think explaining these differences is not that necessary because you already know this, but lead optimization is a very important concept because you have to choose a candidate and then you have to go through the profiling and then you have to come up with the final material. If you think about big pharmas, for the company that I went and worked for, there is a lead optimization process. Usually they use AI to do the lead optimization, but you have to put a lot of time to go through this lead optimization process. But when you do pilot, small molecules itself has a lot of homologous in the kinase. So when you use single kinase, there is not many selective inhibitors. As a result, multiple tyrosine kinesin are the majority. So what does that mean? IDC kinesin is all touched, but because AB's increased in the cancer therapeutics, the toxicity has to be balanced by the efficacy, and it has to offset and outweigh the toxicity. And e.g., a lot of e.g. options are if they have manageable toxicity, even though efficacy is not that greater than toxicity, they tend to be approved. However, this antibody options you do, whether it's fully human life or whether it's humanized mouse experiment, let's say there's lead candidate, and without going through optimization process, they just jump into the cell development. And for cell development, whether Samsung Biologics, Bionex, Wuxi, they all do this area. But I think if you think about our budget, initially you choose the target rate, and then until the first clinical, we put about 50 bill, excluding labor. So, toxicity test and clinical phase 1 test, for example, 1,000 liters of batch production, that will be $3 million US dollars, and then which is 3 billion won in Korean won. So large molecules, the downside is that the pace could be fast, but manufacturing cost is too high. As a result, people are saying, "Oh, then you have to increase the yield." That's why Woosh or other company, they did 2 gram per liter, but now what Woosh is doing, they generate 8 grams, 10 grams, or 5 grams. For our case, we do about 3.5 grams to 5 grams per liter. That's our average yield. And when this happens, depending on the volume of the clinic, we don't know whether 1 batch is enough or 2 batches are enough. If it's going to be 2 batch, then it'll be 7 billion won. So, for a Korean biotech company, it is a lot of cost. It is a big budget, but antibodies and new synthetic medicine, because they have two different targets, you have to go one in hand. They have to be working in parallel. They have to be developed together. It's not like one side is better than the other. And when you're going to treat something inside, that'll be synthetic medicine, and antibody that should be outside the cell. That's the technology portfolio.
And when I got your business cards, there were venture capital people and business workers as well. And when we secure capital, and why the reason why I brought this question up is that you need a lot of money, and when you don't think about money and prepare, then there are cases when even though you advance to a certain point, but you cannot realize that goal because of money. So that is why I brought this question. And among the ideas that you delivered during your presentation, the costs that are linked to developing a new drug is keep increasing. And in fact, in Korea, of course, has been advancing. However, by investing trillions of won into developing a new drug is not easy. So if Korean companies keep working on this, then as time goes by, it is more difficult to raise money. So unlike the past where if you come up with a really good product, do you have to keep waiting and waiting for getting a license? And when you license, and then if you do not want to license out to other companies, then should Korean companies make a union? Should make an association? In Korea, they are raising attention in this bio industry, and how are we going to strategically come up with a new goal? And what do you think, Doctor? Your ideas?
Well, maybe this is my personal opinion, because depending on companies, CEOs have different visions and philosophies, and they have different wills, and they have different ideas. However, in my personal opinion, I think so far, as you mentioned, no matter what biotech company it is in Korea, they need to are struggling to get this funding and also a human talent. If you think about how big pharma works in the USA, when an R&D project starts, before it gets wrapped up, about 150 people get involved, from the discovery to clinical stage 1, 2, 3. I would say 150 come in and then put their hands together and then get this done. So a lot of people have to come in and join the rank. So, as I said, collaboration and open innovation is very important. And among the bispecific antibody therapy. There's IMEC, which is a Chinese company, and we did joint development. And for the last 2 years and a half, this year finally, we are going to do 2 R&Ds in the U.S. That's happening right now. And since a few months ago, we started a new project. So what does that mean is, of course, ABL cannot cover $22 billion dollars, but and also we can't have a human talent on our own. So both companies have to compensate. They have to drive these. For example, do 10 bill and 10 bill, and then you do transfer of the technology, and then we are going to do cost revenue sharing too, 50/50. And by developing ADC technology, we out-licensed this to Siston. And of course, we wanted to carry it on, but as we developed IDC, it cost twice more than what we spent for IDC, and that was just too much money. So eventually, as I said, Morphosis, Genco, just like these companies, when you go work on it over time, it can generate revenues by partnership, by from royalties, and then by doing that, you can do your in-house M&A or development. Then I think in 10 years, these strong, great companies are going to come.
그런 방향으로 가겠다고 정했고요. 저희 말고 다른 회사들은 다른 어떤 방향을 정할 수는 있더라고요.
알겠습니다. 좀 다른 얘기를 하나 드리면 국내 바이오텍, 국내 제약사, 해외 바이오텍, 해외 제약사를 보면 항암제를 개발하는 데가 정말 많습니다. 정말. 그래서 뭐 다른 나라에 있는 건 다 망하고 우리나라 것만 성공한다고 하더라도 너무 많은데 그렇죠. 그걸 보면 다 성공했으면 좋겠지만 결국은 이제 비교 우위라는 게 있을 거고 그래서 우리가 이제 투자자들이나 보통 사람들이 볼 때 어떤 회사들이 항암제를 개발하고 있는데 아 저거는 좀 더 의미가 있다. 저거는 좀 그런데 이런 것들 그래서 뭐 그런 판단을 파이프라인 보고 어떻게 해야 될까요?
그러니까 이제 그 항암제에 대한 그 예를 들어 성공률은 통계에서 보시면 신약 개발의 성공률이 예를 들어서 여기가 9.6, 9.7%면 항암제가 5.7% 정도뿐이 안 되니까 반뿐이 안 돼요. 굉장히 어려운 건데 저는 이렇게 봐요. 그러니까 예를 들어서 제넨텍 하면 거의 브레스트 캔서, 유방암을 거의 그냥 타파했다. 근데 최근에 에스트라제네카는 령 캔서, 폐암을 거의 뭐 대체했다. 그리고 이제 J&J 같은 회사는 최근에 보면은 멀티플 마이엘로마를 거의 대체하고 있는 것 같아요. 그러니까 이제 외국 회사들은 나름 자기네들이 하고 있는 인디케이션에서 굉장히 포커스돼. 물론 그것만 하진 않지만 그 나름 회사에서 이거만큼은 우리가 어떤 회사가 바이오시밀러가 들어오건 신약이 들어오든 혈통같이 지킬 수 있는 강력한 파이프라인을 갖고 있다. 근데 이제 오픈된 필드에서 보시면 요즘 약이 없는 뭐 췌장암, 팬크레아틱 캔서, 프로스테이트 캔서 당연히 이런 게 중요한데 뭐 이런 회사들이 못 만드는 약은 대한민국 회사도 만들기가 어려워서 서로 굉장히 스트럭을 하고 있는 건 맞는 거예요. 이제 저희도 실은 ABL001이라는 어떤 물질을 하나 때부터 저희가 이중항체를 해서 요즘 임상 결과가 좋아서 저희가 뭐 팬크레아틱 캔서에서도 데이터가 좋고 비암에서도 좋고 뭐 괜찮은 데이터들이 나오기 시작을 해요. 근데 이 위암 같은 경우는 외국 회사랑 얘기했을 때 이 매력도가 좀 떨어지는 경우는 있는 것 같긴 해요. 그래서 이 기술 이전을 만약에 전적으로 목표를 하면 이제 웨스턴
So if you're only focusing on technology to focus on indication for foreign people, but in Korea, blood cancer, we do not have that many portfolio, but there's a strong need from foreign companies for blood cancer. So you have to target unmet medical needs and then specify a target, you work on it, and maybe we're just doing a clinical 2 after clinical 1, but once you keep working on it, if you take and embark on this journey, a very tough and difficult journey by identifying indication, there will be a good result.
Thank you. If that is the case for individual companies, then you should take a close look to what overseas or global companies are doing. Well, then you're working on anti-cancer, and what kind of different characteristics should we pay close attention to?
Well, if you think about our company, we do CNS, but other big pharma, if you look at their articles, as CEO changes, their application changes. For example, the current industry trend, CAS was basically buried and then it revives after CEO gets replaced, and NACI or other applications are out there. But even if there are other applications, big pharmas and US pharmaceutical companies are focusing on anti cancer and CNS. Of course, right now they're focusing on infectious diseases because mRNA is leading many different applications, and because there are so many vaccination, infectious diseases were neglected in the past, but because of this, the pandemic. It is emerging as a new trend. So, in my personal opinion, anti-cancer drug, CNS, and infectious diseases. These three are going to be key areas that are be interested by big pharmas.
Thank you. And here comes the last question. And I was kind of confused. Some people approached me last year, and since they know that I was kind of engaged in biospecific antigen, and they say, so probably three antigen. Why not tenfold antigen? They say. But today ADC and bispecific antibody were discussed. So what kind of limitations do we have? To what extent can we extend this? And since we have limited room for labs, and if to make something even fancier, if we say tri-specific antibiotic or so, but in the future, if we take this kind of a multidisciplinary approach, what would be in the future?
Well, whether it's biospecific or ADC technology, if you follow the history of their development, you'll have an idea. When I started my company, ABL, I talked to other companies. I also talked to Hwang Yu, and then when I was doing IR for IPO 2 years later, the most frequently asked question was like, why didn't you do joint therapy? Biospecific has really low yield. It's not that effective. However, I would say that biospecific antibody, the yield, the productability is not an issue at all, because when Hanwha was developing it, it was 1.1 gram per liter. But Bionix, I'm not saying I'm looking down on them, but even Bionix, a Korean company, is yielding 3.4 gram per liter. So productability is already overcame. And as you understand biology, and if you look at all the failures of clinical trials, especially immuno-oncology drugs, biospecific treatment is better than the other option. And there has been accumulated data that proves it. That is why biospecific is considered the mainstream. But if you think about three triple specific, there are companies that are claimed this. For example, Holsetting or Obitoth is added, and then PD-1 is added additionally, and that is making up a three antibody. But there is a productability issue. Like the concept POC works, but the actual production is not at that stage yet. I think in about 5 to 10 years, three triple specific antibodies could be derived, but we have to understand the lesson. What J&J Johnson and Johnson is doing is they are trying to do a combination of biospecific antibody and another biospecific antibody, because 5 years ago, even 5 years ago, people didn't even want to mix up a single monoclonic antibody. But I think if they're now trying to combine two biospecific antibodies, I think triple specific antibodies are going to be generated in the future. When first generation fails, second generation fails, and then now people are talking about third generation. That is why NegoCan and us are creating NegoCan antibody, and we did technological transfer, and for next generation joint development that we are doing with NegoCan, our bispecific antibody is going to be added with ADC. So we're going to go for biospecific antibody ADC. That's the conclusion of our companies. We're still at the early stage. What we would like to do is when NDC is added, we want to know whether a biospecific antibody is going to prove better efficacy than the conventional ones. I think we will need about 2 years to be able to give you evidence or proof. So in that sense, doing tri-specific antibodies, okay, it's fine. But the mainstream right now is bispecific, and the ones that are prepared with this wave will be able to survive and go for the next step.
Well, if you take the real situations, we should keep working on that. Thank you for your answer.
Thank you very much for your discussion. So they have discussed on B-specific antibody platforms for next generation cancer therapy so far. And also they discussed about the challenges that each company is facing in domestic cancer therapy market. So from now on, there will be the company presentations. So first of all, there will be a presentation by Sang-chul Oh, MD, PhD, CEO of Hedgehog. So now I'd like to invite him onto the stage. Please welcome him at a big hand.
Good afternoon, ladies and gentlemen. I am Sang-chul Oh, CEO of Hedgehog Company. I want to introduce my company. I'll talk about the history of my company and some essential core field and some importance of my research topics, Hedgehog pathway in cancer development. My company was built 2018 November. Nowadays, 12 person recruited to the research jobs and some general management. Our company had one research center. I want to talk about some journey to new drug development company. 17 years ago, I was selected one of two international fellowship program at the Anderson Cancer Center. Luckily, I was selected over the Anderson Cancer Centers. At that time, in the lab of GI Cancer Center, my topic is Hedgehog pathway. So, moving to Korea University Medical College, my focus of research topic is only one is Hedgehog pathway. My colleague and I was hardworking to do some research at the Hedgehog pathway. Finally, we are producing so many patents and papers, and we established some hypothesis of the Hedgehog pathway inhibitors. So, 2018, we received some government grant, $2 million US 4 years grant to establish the bio-SPC company. So, at that time, I established that company. Nowadays, our company has 4 pipelines and 2.5 million US dollar government grant and domestic patent and PCT applications. I was supported by Vice President, especially director of a research center, Mr. Hall. He was from the Mogam Research Institute of the green growth pharmaceutical companies. He had a long-standing background of the drug development of target therapy and immuno-oncology therapies. I want to proudly present my company. My company is a very strong point of research-based companies. We are 3 PhD and 2 master degree researchers to the work very hard. So each PhD had one team for new drug development and validations and biomarker studies. Our hedgehog company had several topics of researches. I had an expert of clinical trials. Your family have some gastric cancer. After first-line treatment, you will be recommended second-line Cyramza and paclitaxel, third-line and nivolumab, immune checkpoint inhibitors. I participated in that clinical trials, and I was listed co-author of the Lancet and Lancet Oncology at that clinical trials. Nowadays, I had several clinical trials, first-in-class clinical trials, immune checkpoint inhibitor, and some target therapies. I had some good news for target therapy gastric cancer first line. Maybe next month, that combination chemotherapy was listed Korean FDA for gastric cancer first line treatment. So very proudly, honorably, I am co-author of the first line and second line, third line of gastric cancer standard treatment in the world. And the second one is my research team is focusing on some mechanism of drug resistance and oncogene and specific biomarkers, and very familiar with the genomic analysis of certain cancer tissues. We are long-standing of the collaboration with domestic and international pharmaceutical companies. I received one grant, AstraZeneca 2014. I'm one of the 4 researchers in Korea to receive the grant. The AstraZeneca gave me $0.1 million to grant their drugs to the research. Any other obligation, only research, 1 year, and one seminar in AstraZeneca in Manchester, in UK. At the time, I presentation of my research work at that research center in Manchester. So we had also some good history of the researchers on that cancer therapy. Our company is the very strong research-driven companies. We established one-stop drug development systems. Our company located in Korea University Kuro Hospital, one of 10 hospitals that was a research-driven hospital supported by government. So we can usually easily assess the human material to target discovery. Actually, we had a patient-derived cancer cell, 50 cell lines, to validate our new drug efficacy. And we had some very close and strong partnership in antibody production and small molecule productions with other bioventures. And we had some strong bioassay to define some efficacy of a new drug development. So this process is very cost and toughest work to do some drug development. But we established the one-step, some target therapy and candidate target define and some validations. So very efficacy and low cost and very fast system. This is a very good slide for our company pipelines. The SMO antibody is a post-one product of our company. As you know, the GPCR antibody is very rare FDA permitted drugs, but our pipeline is GPCR antibody and first-in-class hedgehog pathway inhibitors. Nowadays, only small molecules approved for hedgehog pathway. We produce the first-in-class antibody-based new drugs. And our hedgehog pathway had some very strong player in drug resistance. So our inhibitors may be extended some clinical indication for some solid cancer treatment. Hedgehog pathway, I studied 17 years ago. This hedgehog pathway is very important of the malignant behavior of the cancer cells, survival and invasion, migration, drug resistance. So many malignant behavior of the cancer cell involve the hedgehog pathway. Especially, I worked in the colon cancer and gastric cancer. I defined some role of a hedgehog pathway and other pancreatic basal cell carcinoma cell. I want to limit at first the 3 cancer type because of the unmet need. They had some small molecular inhibitors, but some drug resistance. And the colorectal cancer, they had 50% had finally got resistance. So our drug may be overcome this unmet need. So our pipeline like this, first class of the drug is hedgehog inhibitors, and the second one is EphB3, and third one is endothelin antibody. So our first sumo inhibitors driven the mouse antibody to humanized antibody. We already established the RCB. And we had some data showed some control group of small molecular inhibitors. We had a better response in colorectal cancer cells. So we want filing IND 2023, fourth quarters. And the second one, PLE inhibitors showed some better response compared to control groups, but we are now processing of the lead compound. So we had several pipelines, but especially H101 is SMO antibodies. We want IND filing 2023. We analyze market share is like this. So we can overcome some limit of the small molecule of basal cell carcinoma and some drug resistance colorectal cancer and gastric cancer, and no target therapy of pancreatic cancer, so very candidate of our indications. So our business plan is some license out and M&A and some collaboration with the big pharma. So I want to extend my clinical trial to other big pharmas. We had last year we got the domestic patents and we application of the several countries PCT. So maybe receiving good news from near futures. So we want to receive the 20 billion Korean won to IND. So maybe we want to receive the Series A this year, and next year Series B, we want to receive some investment money. So our country is just very small companies, but we are very strong and long-standing of partnership with bio ventures and research centers and to the good drug development. So our strong point of the partnerships. Okay, and thank you for your attention. Yeah.
Thank you very much. We'd like to thank CEO Sang Choi-oh for your informative company presentation. Hope more investors pay a lot of attention on what they have done so far and their vision. All right, then let's move on to the next company presentation. The second presentation of session 4 will be a presentation by Sung-eun Kim, PhD, CEO of Oncovix. So now we have him on the stage. Please welcome him with a round of applause.
Okay. Good afternoon. Thank you for coming here. I am Sung Kim and the CEO of Oncovix. Today I'm going to talk about the anti-cancer drug discovery platform of Oncovix and then its application. But unfortunately, today's my talk a little bit differ from this session's topic, the bispecific antibody. But however, I hope my talk will give you another perspective and possibility of another antibody drug application. So before start, let me introduce myself and my company. I graduated from Department of Chemistry at Gong University and graduated from also the College of Pharmacy at Seoul National University. And then I moved to the United States, and I have worked at the National Cancer Institute, NIH, in the United States as a postdoc for 5 years. And then I moved back to Korea, and then I started my own business 2016 based on those experiences. So since then, my research was focused on the kinase inhibitor development, the discovery and the development. And then I also expanded my the network and then collaboration to the industry, academia, and the institute. Now we have the good relationship and the partnership with our many collaborators, such as JUTH Biotech and Asan Hospital, and then Gyeonggi Biotech, and then the Pasteur Institute. This slide shows about the drug discovery process, as you've seen before many times, but I can explain that in shortly. As you know, to create a new drug, it needs a study for evaluating efficacy and the safety. Clinical studies is a process to confirm whether the drug candidate are beneficial to the human, for the research processes such as basic research and the non-clinical studies. Clinical studies are ordinarily divided into 3 phases, and they performed at the medical institutes such as hospital under very strict regulation for healthy volunteer and the patient. So recently, antibody drug conjugate have been drawn attention. So in shortly, I said ADCs. ADCs are a class of biopharmaceutical drug designed as targeted therapy for the treating cancer. So unlike chemotherapy, ADCs are intended to target and kill tumor cell while sparing the healthy cell. So that means with better potency and then safety and then less toxicity. So ADCs are as of 2019, about 5 or 6 pharmaceutical companies were developing the ADCs. So ADCs are complex molecule composed of antibody linked to biological active cytotoxic payload, like natural toxin and artificial toxin. So Because using the toxin, sometimes ADs raise the safety issues. So as you see in previous slide, part A that shows a simplified general structure of ADCs. Part B shows a general mechanism of the action, the ADCs. So let me explain it shortly. The step one, the ADC binds its target cell surface antigen receptor to form the ADC antigen the complex. And then step two, the leading to the endocytosis of the complex. The step three, the internalized complex undergoes the lysosomal processing. And step four, the cytotoxic payload released inside the cell. Step five, the released payload binds into its target. And then step six, the leading to the cell the death. So now more than 50 different known antigens have been used as a target for ADCs that currently in clinical or clinical development. A few years ago, the Miao group in Ningxia University in China suggests that compared with the traditional ADCs, small molecule drug conjugate, in shortly SMDCs, provide a new point of view, but less established perspective for targeted therapy. In spite of this, SMDCs have several strong points. First, they have non-immunogenic properties. Second, they match more the manageable synthesis, much more than the traditional ADCs. Finally, the low molecular weight with a higher potential for cell penetration, even in solid tumors. So SMDCs might be a promising alternative with small similar efficacy to ADCs to increase safety and decrease toxicity. So now we have some question. Which is more contributes in the SMDCs component? Of course, SMDCs use their appropriate the combination. So among the SMDC component, how can it discover new small molecule cytotoxic payload as a better potency and the selectivity and the safety, the less the toxicity? So at this point, we need some strategies. On this image, along the x-axis, we have two options: the known protein structures and unknown protein structures. The y-axis are the options. The y-axis are known ligand structures and unknown ligand structures. So depending on the combination of these options, we may follow the different drug discovery strategies. The most beneficial is a blue zone. It shows that what can be done when both protein and ligand structures are known. The green zone is next best. When the protein structure is known, but ligand structure is unknown, and so forth. The structure always will accelerate the drug discovery process. So our OncoBix is proposing the small molecule payload discovery strategy. Our platform technology, target-oriented focused for, in shortly, TOPAP, is originates from fragment-based drug design combined with the computer-aided drug designs. Actually, TOPAP has been created using green zone drug discovery strategies. Basically, fragments are small molecule of less than 300 Dalton in size, contrast the larger and the more complex molecule using the conventional high throughput screening. So we properly use the fragment for three main regions. At first, a few hundred fragments can cover the same chemical space, the hundreds of thousands of conventional compounds. Even in a small lab like us, making screening feasible. Second, the fragment can be efficiently measure the protein, the druggable, the pocket, shown in here, the yellow circle. Finally, the development can be readily optimized by growing, the merging, and linking of the multiple hits, whereas optimization of conventional compounds is often very challenging. Instead of using the conventional expensive time-consuming tools like SPR, NMR, isosomal titration calorimetry, we are using the computer modeling. Computer modeling, the computer modeling is easily accessible while saving time and resources. Also, computer modeling makes us easier to understand the binding pattern between fragment and protein. After optimization, hit and read compound, we can propose the effective cytotoxic drug candidate after insights, chemical synthesis, and efficacy evaluation from our collaborator. So this illustration shows a SMDC structure with the applied top-up derived cytotoxic drug in briefly. As a result of applied top-up, the platform to find the drug candidate in 2018, we could suggest an anti-cancer candidate named OBX1 and published the target oncology in International Journal and applied a patent. OBX1 compound identified the highly effective mutant selective EGFR TKI for the treatment of T790M mediated lesions in non-small cell lung cancer. On the left side is a simple flowchart of top-up process, and then right side is a biological data by evaluating inhibitory capability at the cell-based assay in the published paper. The result table on the bottom side, compared with the other mutant selective EGFR TKI, such as almatinib and osimertinib, the OBX compound is a better potency and the selectivity for mutant EGFRs. So OBX112 compound treatment was very highly effective against the human EGFR mutant lung cancer models with or without EGFR T790M, not only in vivo or in vitro, but in vivo. So for example, if we combine this result, we can design SMDCs applied our cytotoxic candidate OBX112 to increase the safety and then decrease the toxicity. Of course, I understand that to develop that drug, we still have a lot of barriers. But I think in spite of this, SMDCs might be promising alternatives with similar efficacy to ADCs. To summarize, the novel classes of small molecules have been developed as a tool for treatment of cancer and other diseases through the different approaches like ADCs, immunotherapy, and then protein degradation. Within the growing the research area, progresses in the ligand screening and the procedure and the hit-to-read optimization. Our platform top-up technology helps to find the cytotoxic drug discovery easier to increase the safety and then decrease the toxicity. And combined with the advances in the various drug release technology, using the antibody. So now we're making the future treatment of the broad range of pathologies with the targeted therapy. The future is right now. So that's all I have to say. Thank you for your kind attention.
Thank you very much. It was a presentation about OncoBix, which aims to develop innovative targeted therapies that meet the unmet needs of diseases that are difficult to treat effectively in modern medicine. So from now on, we will have a short break for about 10 minutes, and then please keep that session 5 will continue from 2:50 in your mind. Right, then we'll see you back. Ladies and gentlemen, may I have your attention, please? In a moment, session 5 will start at 2:50. So we would like to thank all the guests for entering the venue. And also simultaneous Korean English interpretation will be provided for the event. So channel 1 is for Korean, channel 2 is for English. And you can also download the company briefing material files through official website. So ladies and gentlemen, did you have a good rest? Welcome back. So now we will begin the session 5. So session 5 will proceed with a topic of strategies for developing treatment for rare intractable diseases using artificial intelligence. So there will be the topic presentation for session 5. We will have Jin-Han Kim, CEO of Standime, on the stage, who will give us presentation with the topic of strategies for developing treatment for rare intractable diseases using artificial intelligence. So now I'd like to invite him onto the stage. Thank you. Ladies and gentlemen, please welcome him with a big round of applause.
Right. Okay. Can I? Right. Yeah. Hello, everybody. I'm Stan Kim, CEO at Standime. Today's topic is strategy for developing treatment for rare intractable diseases using artificial intelligence. But I'll introduce our AI platform called Standime Ask for you today. That platform is for novel target discovery and prediction. But there is an intersection point with rare disease because rare disease intrinsically is a sort of biological malfunction. And Standime Ask can predict the MOA and the target related things. So we can narrow down to the very genetic level of malfunction or patient level also. So I'd like to introduce our Standime AI Ask platform first, and we can move to the rare disease topic. Right. Standime's mission has three things. First one is value creation, and second one is first-in-class drug discovery, and the third one is continuous supply. First one and second one is very common in drug discovery area, but these two things are very difficult to achieve by artificial intelligence, not the traditional approaches. Third one is very native characteristic of artificial intelligence. Artificial intelligence can generate or predict continuous pipelines and their candidates. Right. So Stendheim has two major AI platforms. First one is Stendheim Eng, and second one is Stendheim Best. We predict novel targets by Stendheim Eng, and then the output of Stendheim Eng is a novel target discovered by Wong for ourselves. Yeah, so there's a very novel target. And then Stendheim Best is for the generating or designing novel compounds. So Stendheim Best to take the output of Stendheim Eng and then generating novel molecules. So we call this process magic workflow AI. And we have very first-in-class assets from these platforms. And we move to our own pipelines. Yeah, these are the modules that each platform has. But we focus on first phase, the Stendheim Eng target identification today. You know, there are so many human proteins, and we know the functions and structures of some of the proteins from the whole human proteins. And some of them, of the known proteins, are used for treatment that we call well-known targets. And there are so many unused for targets for treatment, but druggable for the druggable proteins. We call this area very unknown or druggable proteins. So Stendheim Eng can find that area druggable proteins from the unused for treatment. Stendheim Eng overall map is there are several modules in the Stendheim Eng platform. First one is called Pathfinder. It's actually a composition of two parts. First one is knowledge graph database. We assembled and integrated from the curated external databases and NLP technology for processing the literatures. And there are also, we have algorithms, deep learning based algorithms to infer the target, predicted target, and then MOAs on that knowledge graph database. The other module called Wanderer is omics database data set and algorithms and other modules we have. So Stendheim Eng, as I mentioned, has three parts. First one is graph database. That means this is a very massive database of nodes and edges. Those are all the sort of bio and medical entities, and then the links between the edges are their relationship, like inhibition or binding and so on, or expression. And there are algorithms, deep learning algorithms to infer or predict the relationship, hidden relationship between a disease and target. Some of them we can call the MOAs, and Stendheim Eng has also interactive UI because we cannot solve every problems for drug target discovery because that side is very fundamental and natural science perspective we need. So we have to work together with researchers. So researcher can throw and give a query or hypothesis to the SNIME-esque platform, and they can narrow down to their expected evidence or hint, and finally, they can get to the truth. So yeah, so many nodes and relationships on the graph database, and yeah, we gather the nodes and edges from the literatures in real time, and integrate that nodes and links into the existing our graph database. These are our external databases we use to integrating our own graph database. And this is a very simple example of the algorithms. This algorithm train the patterns of extracted patterns of a target and a disease. That means training examples are their subgraph of a target, a disease. And the result. And Wanderer use very mathematical, actually graphical mathematical approaches we use. So we can follow the protein-protein interactions on the graph database. Yeah, the result. And as I mentioned, researchers can give a query, their hypothesis, and get down to the evidence or truth. We have 11 members now to develop STANDIME-esque platform. 4 members are systems biologists and biochemists, the domain members, and the 4 are AI scientists developing the new algorithms of deep learning-derived inference, knowledge graph algorithms, and so on, NLP algorithms. And AI engineer 3, back-end, front-end. STANDIME has 25 members of AI members inside, so we work together to develop the STANDIME-esque platform. And we have 10 members of pipeline leaders. So we discuss and drive novel targets together in a domain side and algorithm side. We published the results at the RECOM 2020 last year. We did a comparison with GSK's result to the ability of predicting novel target, the hit ratio. And yeah, STANDIME-esque platform is not just anyway, in silico and artificial intelligence predictions. That means just predictions. So we have to move to the validation, wet lab validation with predicted candidates. So we are doing then that validation on these several various diseases, fibrotic diseases, and prostate cancer, and then Parkinson's disease and COVID-19. So one of the fibrotic disease, we got the phase of in vivo, in front of in vivo, and in vitro validation was passed. So we expect to get our own novel target soon, in a few months. Yeah, there are behind the story. We predicted 20 targets in silico target candidate, and the partner pick up 4 candidates from the 20, and one was revealed that already published in the earlier. So we removed one of them, and 3 are selected, and we got a very positive result. All of the 3 candidates. And there are our partners, Iowa Women's University and SK Chemicals, Billa Institute, University of Cambridge, and SK Kids CNC for the double target discovery, not the compound discovery. So we do a role of prediction, and collaborator partner has a role of experimental validation. Yeah, when we work together, we have this collaboration scheme. And I'm scared with the size of the members now. We have over 60 members, and we have 3 members in the US and 1 member in the UK. So in the UK, we work with the Cambridge University, and then also in the US, we have a plan to work together with companies there. And we started in 2015, and from the fund of from the Kakao Ventures, and 2019 SK Holdings, and recently we finished our pre-IPO and from the Invest and SKS, and very recently, a few days ago, we got fund from the Strategic Investment Fund from the SK Chemicals. Yeah, thank you so much. Yeah.
Thank you very much for your presentation. So let's move on to the discussion session. So now, again, I'd like to invite Jinan Kim, a CEO of Standime, on the stage, and Seyoung Jung, the Professor, Department of Digital Healthcare, Seoul National University, Bundang Hospital, onto the stage. So now we have 2 gentlemen on the stage. So when you're ready, please proceed with the discussion.
Good to see you. I'll speak in Korean for the discussion session. I'm Professor Jung Se-young from Bundang Seoul National University Hospital Information Lab. And I'm not sure if you saw this on news, and NHS usually recently authorized this treatment. And it says that you need 2.8 billion Korean won for this single treatment. And the story delivered a story about a child and his or her father. Since it's a rare disease, not many people can benefit from this. But if you actually become a child of someone or a parent of someone, then you are in a dire need to get this treatment. And when I saw this presentation, how you're going to reduce this cost of 2.8 billion won, and if you use 2 different platforms from identifying the target and producing protein. So in terms of rare disease treatment, probably there will be an innovative achievement. So we will begin the discussion in that regards. Thank you, CEO, for your presentation. You just informed a couple of candidates, and your 2 platforms, Target and Discover Protein. And your 2 platforms in terms of developing rare diseases, in what actual circumstances can your platforms can be used, and how are they going to reduce costs significantly?
Yeah, so let me answer you. We have two different stories related to rare disease treatment, and one would be not the STEM but repositioning AI technology that has been pursued since a few years ago, and one of them is mitochondrial abnormality diseases, and we also have another tuberculosis project. And we also have another project with KIST that covers autism. So our focus in such projects would be that we do not target one single disease. We would like to go all the way to the root cause of different diseases. That is our approach. And as a result, that is why we are covering a number of projects. And as you mentioned, Stand Time Ask, this has been going on for a few years so far, but in order for this to be used commercially, we weren't quite ready. But after going through some of the experiments, the results are positive. And as I mentioned earlier during my presentation, just because it's a rare disease, I think, like, let's say another disease has a bigger patient group, or if it's better known biologically, or if it's less known. In the case of AI, artificial intelligence, there's no root difference. Like going or finding, identifying subgroup of a disease is the thing. So let's say we're utilizing substandard mechanism that we have, and I think Standard Mask has a really great point because I told you it's a graph database type, and it's related to that point. Whether it's a genetic level or phenotype level, database integration is available, and as a result, inference is available. And if you put in that data, if you feed that data, and based on certain mechanisms, you might see that certain disease is a rare disease, and you can detect that. And from cost perspective, eventually, it could be the issue of data, or maybe it could be a failure because of. There could be many different failures. The reason why it's not happening is because based on cost and time and energy and effort, ROI is not enough. The amount of money that you can generate is limited. That is why companies do not want to take that risk or take on that adventure. But even before that stage, from biologic stage, if you can get hints and clues, then the later part could be used for gathering forces and serves as a ground. So maybe we will talk about it later, but if you think about mitochondrial project that we're carrying on, it is based in the U.S. And in the U.S., there's a patient group with mitochondrial patient communities. I'm invited there. I also make speeches there, and these patient group collects this data, and then they suggest the demand, and then they say, "Oh, please do research on this area because we would like to know this area." There's definitely a demand. Yes, true. Because it's a rare disease, I think, as you mentioned, patient groups, there is organizations or entities with patients, and there are researchers who would like to overcome their diseases, and their voices have to be delivered to the government and private companies in order for them to respond to those diseases. But you are engaging in many challenging projects, and identifying rare diseases and finding candidates. Even after all these process, if you think about what is happening in Korea, you have to have about 2.8 billion won do you think the Kerry is going to approve this expensive R&D project? So I think if you're working for a rare disease, let's say it's launched in Korea and expected ROI is relatively low, and that is a big risk for your company. So do you have any strategy in order to tackle these different risks?
Well, we by ourselves, we do have our own strategies and other companies' case studies. But to begin, I would like to clarify that my company is not a rare disease company, a related company, but we do have relevant information. But in terms of medicine costs, there are unavoidable issues where the drug costs significantly increase. And in order to decrease that, there are some fast track ways, but I personally think it is all R&D costs. And if one single company just does decrease it just for public reasons and just for research issues, that is not probable. So there needs some kind of fund, either it be government funds or Bill Gates Foundation's support, among other kinds of public aid support, or Pasteur Institute, or other pure science research support. But my company itself, it is also engaging it, but there is a company called Philex. That company, based on AI technology, it works on depositioning of airborne diseases. It finds its sources. And if you visit the website there or take a look at their company materials, then it is almost identical in terms of business planning to our company as well. They do get funding from the government, but basically they engage with a patients community, and the data derived from those patients and the research cooperation is their priority when they conduct their research. And they use artificial intelligence to decrease the difficulty level of conducting that, and they have engaged with various partnership entities for public issues, so for public good. And of course, they are a private company, but the process is rather for public good. Thank you for your answer. So, despite Sandime is not a rare disease treatment developing company, however, in order to come up with a treatment for a rare disease, there needs a lot of attention from the government and the public sector. So, if I understand your answer correctly, so regarding mitochondria related, you just mentioned Hanwei. In Korea, is there any rare disease related company, or is there any channel that raised a voice for their patients? Have you ever heard any voices from those civil organizations, or if there are any, how would they help for your research?
The American mitochondrial patient community knew about us, and then we found about them, and then we had a barbecue party together, and then we just eat together. And they also asked us to deliver speeches and presentations for them, and they also showed that without profit it's hard to be moving. It's basically run by a number of families, and they also asked for some financial support too. I don't know. I don't know what's happening in Korea. I don't know if I just happen to have no contact point, or maybe patient groups are not as vibrant. But I know that there are online based groups and communities so that patients and families can transact different information and data about their disease. But if you think about offline activities and more active advocacy activities, I don't know. If you think about your family's disease, it's curable, but within 5 years or 10 years, you might not be able to cure the disease. But by inviting these companies and listen to their research results and then deliver the results to other different areas or space, then maybe in 10 years or 20 years, that you might be able to benefit other people who are going to suffer from the same disease. That is why active gathering and information sharing is being done in the group.
Thank you for your answer. So far, I have had a discussion session with CEO of Sendime, Kim Jinan. I would like to open to the floor to get questions. If you have any question about the presentation, please raise your hand and ask questions. No question? Thank you for your presentation. I have a question to CEO Kim Jinan. Besides rare disease, nowadays there is a lot of raising attention to microbiome. And at Sendime, do you use artificial intelligence to analyze microbiome? And do you have any partnership or collaboration with other entities in terms of microbiome? Um, I'm sorry to say provide a very simple answer, but no. There are various fields where AI can be adapted, but I think rather providing this answer might help. Actually, in fact, we are not, but I'll just tell you what we are doing as a company. AI is not one size that fits all technology. So, what should I say? Among various fields, there are fields which can adopt AI, for example, data, for example, public data, or other fields where you can verify its results with low costs, and where you do not need to develop a new assay to analyze data. So under the circumstances where those conditions are met, we can use AI. But nowadays, microbiome is a thing. Many people pay a lot of attention, but what kind of information that helps our company in the past is to make it short. In microbiome, I heard that data analysis is really difficult. For example, microbiome in our organs, what kind of patterns do they show? And population is the thing. And if the data exceeds a certain minimum population or not, and making it in a data form is really difficult. So far, when it comes to data or samples, If we try to apply artificial intelligence to microbiome, then it is probably not suitable. That is what we discussed, but I'm not sure. Nowadays, the things may be different, but not sure.
Thank you very much. Let me get a question from the floor. Maybe one more question.
If you have a,
No
go ahead.
question?
If you have a question, feel free to raise your hands and our staff will deliver the microphone to you. Seems like no more questions. So this will end. We will end this discussion session. So thank you for your brilliant discussion time, and they have shared their brilliant ideas with us. So please give them another round of applause once again, and please step down from the stage. Thank you. So they will talk about how the treatment for rare disease can be developed based on artificial intelligence, emphasizing the importance of funding. All right, then let's move on to the company presentations now. So the first company presentation of session 5 will be a presentation by Sung-Uk Lee, PhD, CEO of Arzenomics.
Okay, thank
so
you.
please welcome him with a round of applause.
Thank you. My name is Sung Lee. I'm the CEO of Arzenomics. The Arzenomics is the specialist company in developing the RNA-based biopharmaceuticals. Today, mainly I'm going to focus on our platform technology, and then shortly I want to introduce our R&D pipeline. Okay, so our platform technology is the RNA replacement enzyme called transportation ribozyme. So we try to deliver and express this ribozyme in the format of a DNA vector format. So we try to do the virus as the delivery vehicle. Our company was established about 2017 August, and this is our organization. Now it's about 25 people. So our platform technology is based on the global tetrahymena global intron. And this global intron is naturally perform the self-splicing reaction to splice out itself and then ligate to exons in one molecule. And this intron can be artificially engineered to do transplicing reaction to ligate the two separate molecules in one chimeric RNA. But the original one, the natural one, has the very low specificity and efficacy because the only 6 nucleotide base pairing is enough to ligate the base pairing between the ribozyme and the substrate. So during the last 10 or 15, 20 years, we tried to optimize and maximize this ribozyme to have the high specificity and efficacy. So this is the MOA or our approach. We tried to target the disease-specific RNA, for example, the mutant RNA, viral RNA, or the oncogenic RNA, etc. And then the target and cleave it and degrade the target RNA. And at the same time, we try to transligate the therapeutic RNA, which is attached to the 3' end of the ribozyme, and then we replace it. So the one molecule has two functions. One is the degradation of the target RNA, and at the same time, it can express the therapeutic RNA specifically in the disease cell, which is expressed in the target RNA. So for the first time, we have shown that we have the evidence that this ribozyme is really function in vivo. For the optimization, we try to do first the RNA mapping strategy to identify the most accessible site in the RNA. And we try to modify the ribozyme structure through the P1, P10 optimization and antisense sequence insertion. To make a long story, this approach is based on we try to input the base pairing to bring the substrate and the therapeutic RNA together in the spatially. So this slide shows the feature of our platform technology. As the other RNA approach, our approach has also can target the undruggable target because we target the RNA. And as I said, that it has one molecule has the dual function. And also we express the translating RNA dependently on the level of the target RNA. It means that we can regulate the therapeutic gene expression, and using the DNA vector, we can maintain the gene expression. And we try to do the gene editing at the RNA level, not DNA level. It means that we can minimize the genome toxicity and the external genome the change. And we had to maximize the efficacy and specificity through the modification. And also this library has a very high fidelity, and there is a low very off target. And I want to also notice that this ribozyme can function itself. It means that we don't need an extra protein, and we don't need any kind of the inherent cellular mechanism to function. And also the substrate RNA binding site and the therapeutic RNA site is the structurally separated. So it means that we can do easily modular engineering. It means that we can expand diverse indication. So our approach is that to explore the development of the transferable ribozyme-based therapeutics against the indication with a highly unmet need, medical need, in which these unique features can be the most competitively applied. So RNA pipeline. So this is our RNA pipeline and the milestone. We have the RG001, 2, 3, and 4. And 1 and 2 is against the cancer. The 1 is against the HCC, 2 against the glioblastoma. And 3 is against Alzheimer, and 4 against the retinitis pigmentosa, which is a rare genetic disease, retinal genetic disease. RG001 is the leading pipeline. And now we are manufacturing the GMP production, and now in the middle of the GLP toxicity study. So we are scheduling the IND application to the US FDA and MFDS end of this year. And second link pipeline is RG004. Recently, we selected the candidate molecule, and we try to do the manufacturing from the end of this month, and we try to do IND application into the US next year. So RG001 and 2 is based on the adenoviral vector, and 3 and 4 based on the AAV. RG001 is against the HCC, and our approach is against the target, the HTALT. HTALT is as known, the most of the cancer cells, and also the HCC, including HCC. So when you compare the another approach, targeting telomerase only, our approach has more advantage because our approach has two functions, as I told you. First one is we inhibit HTALT to inhibit the telomerase activity, and at the same time, we try to express the therapeutic gene activity in the cancer cells, specifically cancer cells expressing the TALT. So for the therapeutic gene, we use the herpes simplex, the virus thymidine kinase, which has the bystander effect. And we select the adenoviral vector after we screen the lots of different kind of the vectors, the viral vector, and then we select the adenoviral vector is the most proper vector system for the HCC. So for the optimization of our ribozyme, we try to do two different stages. One is we try to modify the ribozyme itself, as I told you in the previous slide. And second approach is we try to optimize the expression cassette to express the ribozyme specifically and efficiently in the cancer cell. For example, we try to use the splicing site at WPR to enhance the post-transcription expression of the ribozyme, and also we try to do microRNA target site to regulate the gene expression, specifically in the cancer, not in the normal liver. As I told you, we select the most optimized one, the candidate molecule, which is called RG001, and this is the efficacy study in the intrahepatic multifocal the xenograft of the model, the single dose, and then very highly efficient inhibition of the cancer. And this is the non-GLP the toxic study. And also we have the BD study in systemic injection of the ribozyme. And as the delivery route, we select the intratumoral or the hepatic artery route, because the hepatic artery is, as known, is the main blood supply route for the HCC. So this is the data for the biodistribution data when we inject the hepatic artery injection into the rat. And this is a rat cirrhosis associated HCC model. And then when you compare the intrahepatic, the hepatic artery route, and then also the systemic route, and as you can see there, the adenoviral vector can be more efficiently delivered to the HCC when we deliver into the hepatic artery. And this is the mechanism of the ribozyme. This is the telomere-dependent the mechanism, and this is the telomere-independent mechanism to inhibit the cancer growth. And most importantly, also when we inject the ribozyme in this syngeneic model, then there is very highly efficient immune filtration into cancer. So this the mechanism shows that we are trying to do combination therapy with the ribozyme and with the immunotherapy. This is we also screen the HCC patient with the most applicable indication. So as I told you, now at this stage we are now the GMP production, the GLP tox study. So end of this year, we hope that the IND application into the US and the MFDS. We try to do IND in the combination of the phase 1 and 2A. And also, as we can expect, the ribozyme can work with all kinds of cancer cells, especially the GBM is the most proper, the most effective the indication for our system. So we try to expand our system into the GBM. For the GBM, we also try to do further modify our ribozyme system to express the immune checkpoint inhibitor single chain antibody for either anti-PD-1 or anti-PD-L1. So we can try to express this kind of the immune checkpoint inhibitor combined with HSV-TK tumor specifically. Then we show that in the case of GBM, this case we call it the RG01 plus is more efficient than the RG01, and also RG01 is more efficient than the combination therapy with the RG01 and the immunotherapy antibody in the case of the GBM. So RG003 is against the Alzheimer's disease. For the RG003, we try to target ApoE4, which is known as the highest risk factor in the Alzheimer's. So we try to target ApoE4 and degrade it, and replace ApoE4 with the ApoE2, which is the benefit factor. So we try to AAV and then we try to deliver into the intracisternal. But this is the early stage, and we try to selecting the candidate molecule right now. So RG04, the final pipeline, I think that this is the most proper, most competitive in the indication which can present our unique features of our ribozyme system. RG04 is against especially the autosomal dominant RP. So in the case of the autosomal dominant RP, we need the two functions at the same time. We should reduce the dominant phenotype, and at the same time, we induce the wild type phenotype. So as I told you, our mechanism is the two functions in one molecule. So the autosomal dominant phenotype is maybe the most proper the indication for our system. Among the autosomal dominant RP, we try to target the rhodopsin. Rhodopsin is the most frequently found in the autosomal dominant. But that is very difficult to treat the rhodopsin mutation because, as you can see here, that there is about more than 150 different sites in the patient. All of them is the coding region. So it means that we need the therapeutic molecule which can target all kinds of different mutation in one molecule. So we try to do 5' UTR as the target site, and fortunately, we can identify the most accessible site in the 5' UTR through the RNA mapping strategy. So our approach is to try to do the rhodopsin mutation and degrade it, and replace the rhodopsin mutation with the wild type rhodopsin. So in a single molecule, it can do the dual function, and we try to do AAV, and then we try to do subretinal injection. So we have already selected the candidate molecule which can efficiently restore the vision function in the mouse disease model. So We hope that the next year we'll try to do IND application. This is our IP listing in our company. And finally, this is our development strategy in the next 3 years. We are hoping that in the 2023, maybe the whole pipeline is in the clinical stage. Okay, thank you for listening.
Thank you very much, Sung Lee, the PhD CEO of Argenmix. You may step down from the stage. Thank you very much. And we have an announcement to make to the audiences on site. So for safe event proceedings, we need to follow the government's guidelines to prevent the infectious diseases. Right, then let's move on to the next presentation. So next will be a presentation by Hoon-Taek Kim, PhD, MBA, CEO of Tium Bio. So now we have him on the stage. When he's ready, I will introduce him once again. Now let me introduce once again Hoon-Taek Kim, the CEO of Tium Bio. Please welcome him with a big hand.
Good to see you all, ladies and gentlemen. I'm Kim Hoon-Taek, CEO of Tium Bio. I was educated in the States, but since I do not speak English well, so I deliver my presentation in Korean. And it's been a while seeing all of you, and it's been a while introducing my company. So thank you very much, all the staff members here, for giving me an opportunity to introduce my company. So here's an overview of my company, and we will deliver on the fibrosis treatment opportunities and our vision and our plans. Our company was founded in December 2016, and with our R&Ds and our researchers. So we're a new drug company, and since the third year, we went public on Kosdaq, and that tells that we are competitiveness, and our research competitiveness is verified, I would say. And so far, there were technology transfer, 2 cases overseas, and we have 106 patents and working on business. And last but not least, for this slide, even though we're located in Korea, but in August last year, we collaborated with Moyum, and we made that a subsidiary company of ours, and 11 staff members are working as researchers there. And in Australia, we founded another subsidiary company, and it works on clinical trials. And based on that, they continue working on their clinical trials. And the key focus of our company at first is endometric hemophilia. And hemophilia may sound very new to you, but there are cases. It is a very serious disease. It can make you lose your life if it becomes severe. And hemophilia treatment is I myself and other key researchers, when they used to work for SK Chemicals, they worked on R&D, and in 2019 we got CA licensed out, and in 2016 we got approval from FDA. So it is the first time ever in Korea, and nowadays we are saving lives of people all around the world from 40 nations. And it was also listed in the Korean health insurance system, so Korean citizens can get support when they treat hemophilia. And next is anti-cancer. And I may not be able to cover this part, but TGF or BGF receptor 2 is also being worked in our company. And nowadays, immunotherapies weaknesses can be covered thanks to this technology. So we are currently in progress. And also for immunotherapy pipelines, we're currently working on that as well. And next is fibrosis, which is the key topic for today's presentation. And actually, back in 2018, we have been working on this technology, and we made a technology transfer, and we worked on IPF, and we treated this, and we are taking a key focus on this. And next is that NASH trash treatment, and in 2020 last year, we got support from Ministry of SMEs, which was 2020 Big Three project. We were designated as a company for that and currently working on R&D. Well, then, let's move on to the key topic for today, fibrosis. So what is fibrosis and why does this happen? I will begin with that. If I would define fibrosis, then fiber is accumulated in your inner organs, and that disease is called fibrosis. So fibrosis, the definition of which and the term itself is really difficult. I myself is an expert, however, for Bai people, probably this term may be really difficult. But to put it simple, when you make a wound, then there will be some kind of inflammation, and then you will have a scar later on, and then blood will be solidified. And so they are formed with tissues, and those combining tissues. But if let's say if your inner organs and or livers and if you get scars, and if those organs cannot function properly, then it can later on potentially make you die and lose your life. So for those serious diseases, they are defined as key reasons why people die, and for IPF, stands for idiopathic pulmonary fibrosis, and there are also NASH. And however, nowadays the treatment for those are not still yet to discover. But if you take a look at the market growth rate recently, it is a market where it is gaining a lot of attention. Next is the process of getting fibrosis. So if you take a look at the slide, usually when a normal organ gets damaged, then the cells will die. And in order to deal with it, inflammation happens. And when inflammatory processes proceed, then later on, if they are healed, then they can turn normal. However, if this process repeats itself, then it can begin fibrosis. So what I want to say here is that when your cells or gets wound, and if this fibrosis response happens, and if you memorize this part, then it'll be easier for you to understand what I elaborate in the other slides to come. And next slide, there is NASH treatment. And we got two treatments of IPF, and for NASH, we do not have an approved one yet. But the processes that I mentioned before, globally, there are a lot of treatments under development, and to which parts they relate to, they locate and is shown on this slide. And for IPF, anti-fibrosis, there are treatments. We are highly focusing on developing anti-fibrosis. And for NASH, as you can see on the slide, unlike IPF, you can cause this liver cells to die. There's metabolism, and in order to control this metabolism, there are a lot of different drugs on this part, as you can see from the slide. And next is IPF treatment. In order to come up with an IPF drug, for us, we have been working on developing a fibrosis treatment for 20 years, and based on our clinical data, and we wanted to see what kind of efficacy these different pieces of research has in terms of clinical study. And we studied commercialized drugs, and we studied this, and these suppress the worsening of these processes. And if we take a look at those failed drugs, and in terms of litonin and pirfenidone, there are a lot of efficacies. However, if you take a look at the failed drugs, then they only suppress specific characteristics. So in order to use it for IPF, then we should highly focus on fibrosis. But so in that terms, thanks to the technology transfer that I just mentioned, thanks to that characteristics, and we can suppress MP5 expression among others expressions. So thanks to that, that is why we could successfully make a success for technology transfer. So based on that analysis, what our plan is that the conventional IPF drug, in order to come up with a new one, there are a lot of global pharmaceutical companies that have been working on developing this, but we also have to come up with a new drug for this, and that is our plan. And what our plan is that if we use only one drug to target various targets, or another is that we suppress one target, but at the same time can mitigate other agents. So we take that strategy into consideration. And of course, recently there is a trend where those conventional drugs and new drugs are used as a combination to treat IPF. There are a lot of attempts, so I would like to mention that as well. And next is our plan to develop a NASH treatment. And NASH, when a liver disease occurs and then there's inflammation and it worsens and it moves on to cirrhosis. And a lot of biopharmaceutical companies are working on developing this. And on top of that, we also work on anti-inflammation and anti-fibrosis. So we have to develop that drug. And in order to catch two birds with one stone, we have to select and secure those characteristics, and that is what we are currently working on. And at Teum Bio, we have 9 pipelines, and step by step, we cover each step and see and analyze and which specific steps those drugs can work on. And we are currently developing drugs. And there's a specific item called TU5113 or NCX003, and it can propel the treatment function, and it also has a suppressing function. So it has two dual functions, and it can work on diabetes, and it can suppress inflammation, and it can suppress fibrosis. So overall, that is the overall result, and so we're currently working on developing it as a NASH treatment. And this agent called TU5113, how much potential does it have can be shown here. And currently we're globally developing this, and it has excellent results. And based on our preclinical trials, when we worked this, even if we, with a very small dose, the efficacy was really good, and it has 100-fold of safety. And of course, in terms of PK, we orally administered this and found out that we can treat NASH as well, which is very meaningful. In Korea, there are a lot of drugs under development, and it is limited to injection. However, there are oral pills which can augment the treatment. And here, during the clinical trial, we decreased the glucose level, and then we increased the peptide level. And here, what to take note is that you may have heard of anti-fat disease pills or drugs, and that itself by increasing its dose in the human body, you can come up with a lot of treatment level. And even for anti-inflammation, we also came up with fruitful results. And Tium Bio here, we are putting a lot of efforts to come up with this fibrosis treatment. And we have compound efficacy test system as well as medicinal chemist team, and they're highly collaborating with each other to come up with fruitful results. And there are two different tools for antibody and small molecule, and they work on antibody, new drugs. And in order to effectively come up with a development, we're in collaboration with GenScript. And the target that we secured here is that thanks to our nanoclicks, and we provide onco design or ChemDiv, among other companies, are in collaboration with us. And this is the last slide. And actually, I just said, besides the fibrosis treatment, here there is endometriosis and treatment. Currently, we just working on the second clinical trial, and in Korea, Daeung Pharmaceuticals is developing this and working on the second trial. And here, TZ beta and HF receptor 2 is currently under development. And for this, in the United States, for the first clinical trial, we are planning to apply for the first clinical trial in the US FDA. And the technology transferred ideas are keep under development, and the rest of fibrosis treatment are under discovery planning stages. And within this year, we are planning to complete preclinical trial, and next year we will begin clinical trial to move on to further stages. And for hemophilia treatment, we are taking diverse steps for this. And in Korea, I personally think that we're at a top tier position, and also in order to secure the top tier position globally, we have strengths, and in results, we will additionally act as a paradigm shift maker. So Kium Bio does have a secured pipeline. And lastly, Team Bio is keep making a growth, and how we made this potentials and growth is thanks to all the supporters who were enthusiastic um, about our company. So I urge you to keep sending your support, and thanks to your support, we can actually work with a lot of stakeholders and make a success in Korea and globally. Thank you for listening.
Thank you very much. It was the presentation by Hoon-taek Kim, the CEO of Tium Bio, a world-recognized R&D company for creating market value through innovative new drugs. So last but not least, I'd like to invite the last presenter, who is Il-jin Lee, the PhD CEO of D Nature Labs. He will introduce his company from now on. Please welcome him with a round of applause.
Good afternoon, ladies and gentlemen. My name is Kim Il-sin from Denature Biolabs. Because it's a new company, I decided to promote our company in Korean so that it can be more accurately delivered to you. We have neurodegenerative diseases, therapeutic approaches, and I'm going to talk about the present and the future of our company. Before I go into the presentation, then please look at the disclaimer. You can see that Denature Biolabs is a total brain healthcare bio R&D company. We not only cover neurodegenerative diseases, but also total neuro-related diseases, neural stem cell platform. As a therapeutic, we use this platform so that hard-to-cure diseases, neural diseases, and disease-specified therapies are created. And in particular, from neurodegenerative diseases, we are focusing on strokes and Alzheimer's diseases. And as an aggressive marketing event, are trying very hard to introduce a neural cell therapy into Korea, TAURX, and we have shown many great results. As you can see from the graph, global drug market is showing 7.2% of growth, and by 2026, it'll grow to $62 billion. And you can see that stroke and Alzheimer will take up about more than 50%. And here, orphan drugs are going to account for 6.6%, including ALS and autoimmune diseases and multiple sclerosis, maybe taking up about 1%. And there's a steep growth, which is 22% on annually, and about a 76 billion-dollar regenerative disease market is going to grow to 76 billion, as I mentioned, and once stem cell therapy market that can apply our direct technology will amount to $13 billion by 2026. And also Pinta neural stem cell is different from the conventional stem cell therapy. Compared to the conventional ones, it is going to show off better efficacy. You can expect this, an membrane stancer or MDSU option. Compared to those two options, it also boasts better safety. And for cell source, we are using first trimester neural parts. We take stem cells from the first trimester fetus, and then as you can see from the images, there are neurogenesis stem cells, and they create neurons. And during the second or third trimester, they create glicogenesis. So if you look at our platform, it is divided into different levels. First, we use fetal nerve system, and we culture stem cells, and we get as many cells as possible. And once these cells are collected, then each neuron cell is going to be utilized for the right condition, and the final products are collected. And these final products can be placed into pathophysiological feature, and that's the platform that we have developed and have been utilizing. The first pipeline that we have is to target ALS, which is called Lou Gehrig's diseases, amyotrophic lateral sclerosis. This is the most common motor neuron disease, and once it breaks out, then patients tend to die within 5 to 10 years. It's a rare disease, and the current options, medicinal options, are relatively slow the progress of the disease, but there are still existing unmet needs. About 5.6% of CAGR is expected over the next few years. Not only that, if you look at the domestic market size, about 4,500 patients are expected, and the domestic market is going to be around a 50 billion won market. As a result, Bio Labs is going to utilize, will develop an orphan drug, and for MOLE would be motor neurons when they're dying out. We have to improve the environment so that more motor neurons are not dying out, and they for the GDNF secretion, we can get the disease or treat the disease. And another possible applicable disease would be stroke. Stroke is basically where you develop a blood clot, and then that eventually kills the cells related to that region. And that environment, for that environment, you can see there's TPA. It's the name of the medicine, and you have to administer this medicine within 3 years. That's 3 hours. That's the only option to cure stroke. Aside from that, there's no available options. But because the society is aging, the market is growing rapidly, and in Korea, more than 620,000 patients are expected, and the market size is expected to grow as well. So that quickly administrable allergenic neurocell therapy is being developed, and this cell therapy, what I can tell you for now is that in the case of stroke, the first reaction is immunoreaction. So because it's an immunoreaction, neural cells can exude different materials, and that is why we would like to target that very factor. So for the first year of R&D, we are going to develop cell therapeutics, and we need to make sure that we have a large-sized, mass-produced cell base. And then the second year, utilizing cell therapy, we have to have GMP facilities in order to create cell banks so that preclinical cells are generated. And by utilizing preclinical cells, we have to carry out non-clinical animal tests so that toxicity and any other chemical medicine producing procedures can be adopted. And then from third year, we're going to collect documents and materials for cell bank and animal tests so that clinical studies can be embarked. And currently, clinical data cells that can be produced in GMP is very important. The initial creation, as you can see from this image, we're focusing on AMD and stroke. But once the cells are created, after that, you can also target multiple sclerosis, HIE, and other neurodegenerative diseases. And I think the scalability is going to be quite sizable. Next, I'm going to share open innovation project. There is LMTX under Taurex Therapeutics. This LMTX is going through clinical stage 3 and 4 for Alzheimer's disease treatment. And you can see this image on the right side, very complicated looking poster, and Tau or X has been accumulating Tau platform for a long time, and with this established platform, for the first time in the world, Tau-based Alzheimer's disease treatment was developed, and it's going to become a game changer in the relevant industry. So now I'm going to look at pharmacologic principle of Tau therapeutics.
Neurons which communicate using electrical and chemical signals. Through binding at its supports microtubules needed for cell structure and to provide rapid transit pathways for vesicles and cell components to the synaptic junction. Aging brain neurons lose some ability to clear waste material, which can accumulate as lipofuscin, where tau can bind and misfolded tau can capture normal tau, causing it to misfold and form oligomers, which disrupt the synapse. Tau oligomers can transfer to other neurons and initiate the same process of tau capture, causing the disease to spread. As oligomers aggregate and grow, they form paired helical filaments and then bundles called tangles. As neurons fill up with tangles, they can no longer function eventually burst, leaving behind just tangles. The experimental drug LMTM prevent tau aggregation by binding tightly within the misfolded repeat domain, and actively releasing individual cell These are shunted to proteasomes for degradation. Cleared of tau oligomers and synapses can work properly again. LMTM therefore has potential to slow and stop disease progression and preserve neuronal function.
Yes, related to Tau or X Alzheimer's disease, I'm not going to reiterate this twice, but according to the domestic materials, if you look at top 5 medicine lists, the global market is targeting Alzheimer's disease and I think dementia. Targeting dementia has a very big social and economic impact. As you also see from here, Early this year, avucanab was partially approved, and this is now becoming a hot topic in the dementia disease industry. And for cognitive disabilities, because it's not fully approved, dementia or Alzheimer industry is waiting for a new innovative drug, and there are ongoing unmet needs. And recently, the interest for tau is going up. Instead of amyloid beta, tau is gaining attention as a root cause of Alzheimer's disease. And as you can see, if you look at clinical part 3, tau RMS, LMTX is the only material, and there are many different tau materials as candidates, but tau LMTX, because it is going through clinical stage 3, it is a leader compared to other options, and because it's antibody-oriented mechanism, when it comes to cost, it has a clear advantage as well. That is why for the in order to properly import and have the right to distribute tau in Korea, we made and signed an agreement, and now it is going through the clinical stage 3 called Lucidity. Lucidity went through 2 different rounds of clinical study, and based on the data, it marked and specified candidates, and then it also came up with efficient dosing. That is why I think there is a high chance of success. And the conclusion of Lucidity is that the top line result is going to come out early next year, and then late next year there's going to get approval for new drug from FDN and MFDS. As you can see from this image at Denature Biolabs, we are going to claim the right for the distribution, production in Korea, and also partial IP in Korea, and also clinical study right for Korea as well. And we're now going through that whole process. Not only that, during the pandemic, LMTX, we wanted to see whether repositioning is available, and there are preclinical data produced, and we saw a possibility. As you can see from this image, in Iran, there were phase 2 and phase 2 clinical trial that went through, and you can see the oxygen rate went up, and the breathing got improved as well. As a result, Tau RX and our company are going to plan clinical studies targeting severe coronavirus patients. And as you can see, LMTX production, domestic production, distribution, IP rights, and lastly, clinical trial right will be handed over to our company, and it is being processed. This is everything from my side, and because we are a relatively new company and I'm not so experienced, but please stay interested and give support to our journey in the future. Thank you very much.
We have listened to the very promising company, which is Denature Biolabs. I'm looking forward to seeing the better future of Denature Biolabs. All right, we would like to thank all the speakers who participated in the presentation and a discussion session. So until we have had 3 sessions today, session 3, new therapeutic strategies for NK cell targeted immuno-oncology. Session 4, B-specific antibody platforms for next generation cancer therapy. Session 5, strategies for developing treatment for rare intractable diseases using AI. So we have had discussions and presentations on these 3 issues, which allowed us to share mutually beneficial information. And I hope that today's event will help promising domestic companies enter the global biohealth market. So this brings us to the end of the second day of BioKorea INVEST Fair 2021. The BioKorea INVEST Fair 2021 will be also conducted tomorrow from 10 a.m. with a topic of session 6, Global Business Strategy in Medical AI Industry, And session 7, Hospital-Centered Biomedical Cluster and Industry Academy Collaboration Model. Furthermore, you can watch the presentation video again through the official website of BioKorea Invest Fair 2021 until June 21st. So please pay a lot of attention until the end of the event. So once again, we are very grateful to you who attended today's event out of your busy schedules. We'll see you tomorrow. Thank you.